This presentation is on life and fossils, and it's actually the record of evolution in the rocks, and actually the fossil record. As we begin here, I want to give you a little bit of background first on the on the biology of fossils. Species are the fundamental unit of biology, and so each species can interbreed with other members of its species, and they must have viable offspring. So that's the definition of a species. Now, a species is actually given a name, and so it was Carl Linnaeus who came up with this, Carl von Linnaeus. He was a Swede, and he gave himself the Latin name Carolus Linnaeus, and so Linnaeus is responsible for the Systema Naturae, and that is the dealing with the classification of animals, plants, and every other thing pretty much on Earth at that time when he wrote this back in the 1700s. Today, we not only use his classification, which is called the binomen, which includes the genus and species. So if, for instance, we were to name the human species, we would say it's Homo sapiens. And so it has two parts to it, so hence the name binomen. And the two parts, of course, would be the genus and the species after that, indicating that, of course, a genus can have more than one species within that sort of group. That is a conceptual sort of idea, however. So, genus, there's not really natural, we recognize relatedness between different genera, which is the plural form of genus. But we do not recognize it. It's not everything within a certain genus will be interbreeding. And so it's just the species that are. And so the binomen consists of those two parts, right? So the genus and the species together. Oftentimes they abbreviate the genus if it's well known what it is. So for instance, for Homo sapiens, you would say H period sapiens. And a few rules to keep in mind about this, actually. One of them is that we always put those sorts of things in italics. And so you'll always see Homo sapiens with a capital H and a lowercase s. And so Homo sapiens is the name of our species, and that's the way it's written in italics, just to indicate that it is a species as opposed to being just some other collection of two words. That is the binomen. So Homo sapiens, and I try to follow this in this presentation as well. Irvingella major is a trilobite. That's another example here, and you can see that it's in italics. The Irvingella, the I in Irvingella, is underlined or is capitalized, and major, of course, is not. Sequoia sempervirens, that's the coast redwood. That is another species. Okay, so the Sequoia sempervirens is a species as well. So capital S for Sequoia, lowercase for sempervirens. These rules come from the International Code for Zoological Nomenclature, and even fossils follow these same rules. And in fact, even ichnofossils, which are related to trace fossils, follow the same rules. So what constitutes separate species? They have to be interbreeding, and they also have to have viable offspring. And so that's the contribution that Carl Linnaeus gave us. He didn't give us the definition for a species. That was developed much later, but he at least gave us this classification scheme for recognizing what a species is and how we can use two names to describe it. Now, given that, there's also a sort of hierarchy of names that we apply. So if the species name, which consists of the genus and the species, okay, so you can see those two at the bottom down here, that's how life evolved, okay, through species, through speciation. There are other concepts that we use as well, and we look at the relatedness. And so when you classify things, you say, well, that's kind of related to this. And so, for instance, if you had nuts and bolts and you had nails and staples and things like that, you would say that those are all fasteners, right? So they all belong to a category of fasteners. And so the same thing applies to species. So if we have several genera that seem to be related to one another, they may be placed within a family. To give them an idea, to give biologists an idea, or evolutionary biologists, taxonomists, and systematists, they actually is what they refer to in biology. These folks have a better understanding than about the relatedness of different genera. Those are placed into a family. When you have several families, you can place those into an order. When you have several orders, you can place those into a class. So some examples we're going to have in the next slide, in the next couple of slides here, but phylum is the next step up from that. So phylum is kind of like, the plural of that is phyla, And that's kind of what we look at when we just describe, oh, what kind of fossils do you find in this rock? And say, well, I found brachiopods. Well, the phylum is Brachiopoda. I found some gastropods in there. Well, gastropods are actually a class of mollusca. So mollusca is that phylum. Kingdom is the next higher order above phylum. And then, of course, above that, you can get domains. And above that, actually, some people use the term realm to describe the relatedness of almost everything, right? So I think there's like 3 realms today that are recognized. I think they're back in the 1980s when I was studying this in your shoes, I guess there were 5 kingdoms that were recognized. So today it's expanded beyond that. We recognize that today the Archaea are one of the major forms that we have. And so that in the old days would have been included in with, they called it archaebacteria, so ancient bacteria. But those are the main ways that we classify things. And so you see here, kingdom, phylum, class, order, family, genus, and species is the traditional one, and domain they've added to the upper end of that. And so the binome and how we describe things. On to the next slide here. This is an example. So Eukarya are things that have eukaryotic cells. They have a cell nucleus. Within the group of animals that are eukarya, we have animals. And we also could have plants, or we could have fungi, or we could have protista. And so here you see the at least 4 of those kingdoms right there recognized. So in all, there's like 6 kingdoms. Okay, those are the ones that are numbered here. And the other domains are like the bacteria and the Archaea. So Archaebacteria is now a kingdom within Archaea. So that's just an example of how we break out the upper end of the classification scheme for biological organisms. So if we just took a dog, let's say a dog. There he is down here at the bottom left here, right? That pug. So he is regarded as Canis Familiaris. Okay, it's the family dog, if you will, or the familiar dog, right? So the Canis is the genus that dog belongs to. If we looked at other canids in the family, Canidae, there's another genus that would cover things like coyotes. And so coyote belongs to the genus Canis, but it's a different species, okay? And then if we looked at like Canidae, we also include a genus that would include, I think it's called Vox, I think it is, or let me think what it is. I don't recall what it is, but the fox is actually a canid, but it is not the genus Canis. And so, but it is related, of course, to the dogs and also to the coyotes here. And so the Canidae would be that. And carnivores, anything that's carnivorous, right? The carnivorous animals would include a badger, which would also include a dog and a coyote and a fox and so forth. But it's a different family than what the fox is in the fox. And so the fox and the coyote and the dog are all related within that same family, but it doesn't include the badger. So the badger is the outgroup for that group then. If we just said Mammalia is all mammals, right? And so including human beings, right? So here's the giraffe as an example. He's an outlier for the carnivora. So giraffes are not carnivorous, in other words. If we take the next level up, we go to the chordates. And so, of course, a giraffe is a chordate, and all the other animals below are chordates. But so are fish. And so fish are like one of the stem groups from which all of Mammalia sprung. And so fish are chordates. Some fish are chordates, not all of them. No, hold it, they are. Yeah, they are. Some of them are actually made out of cartilaginous fish, however, so they're a slightly different category than the osteichthyes that we're familiar with. So like the largemouth bass has bones in it, right? So phosphatic bones. Whereas the sharks don't, but that's a different type of species. And so, but they're both chordates. And so they have a notochord that runs through the backbone. And so even if you have a cartilaginous fish, they're going to have a backbone as well. Above that, then we have the Animalia, which would include things that don't have backbones, like the jellyfish. And then even above that, the eukaryotes, or just anything that would have, in fact, a cell nucleus in it. So that is a classification of the common dog, but it's the relatedness of all other living organisms that belong to the Animalia and also the Eukarya. So you get that idea. So here we have a human being. So there's Mr. Bean down here at the bottom left, and you can see that he is related, in fact, to this other genus of Homo, I think it's Homo heidelbergensis there, and so they're both a species of the genus Homo, and you have the genus Homo that would include several animals that would be part of the part of the hominids, right? So the hominids are a family, but that also includes Pan, the chimpanzee that's over here on the right-hand side. And so those are all hominids at the lower part there. And then above that we have primates, which would include also the baboons. So the baboons are an outgroup or the stem group from which all primates came. And the hominids belong to the primates then. Of course, we're back in the Mammalia after that, and then the Chordata and the animals in the Eukarya after that. So those are just a couple examples of some species and how they're related to one another. So in other words, when we use biological classification schemes like this, we are actually trying to look for relatedness. And it's all based on different models about how we go about trying to show relatedness to these animals. Cladistics is the number one way that people do this these days. It was developed in biology. So we look at shared common ancestry. What are the characters that certain things have that are in common with other groups of plants or animals or fungi and so forth, right? So when we look at these sort of groups, if there is a whole bunch of different groups that will have that shared common characteristic, we like to say that that is a monophyletic group, one single phylum, one single tree that would allow those things to all be classified together. Sometimes we're not so lucky, however. For instance, if we look at warm-bloodedness as being a characteristic, well, it's not just the mammals who have warm-bloodedness, so do the birds. And so aves are an offshoot of course, reptiles and so forth. And reptiles probably gave rise to the mammalians as well through the amniota. And so that's what we share in common with many of the reptiles. And so you can see here that in this figure that's in the on slide number 6 here, you can see that that's considered to be polyphyletic. So if we just took that one characteristic of being having homeostasis, of having warm-bloodedness, that then we would have a polyphyletic group. Sometimes we have paraphyletic groups, which we have some of the organisms within that branch would share that common characteristic, but not all of them perhaps. And so that would be paraphyly that's listed on here as well. And so that would include things that in fact aren't warm-blooded within the reptiles. So paraphyly. So we try to look for monophyletic relationships between organisms when we do that, the classifications. And so that gives us a sense of the evolutionary tendencies of these animals to have certain characteristics. Sometimes we may develop, and for instance, if we just took things that fly, right? So things that fly would include mammals, right? So we have bats, we have flying squirrels that more or less are on a glide path, but certainly bats fly through their own propulsion, and so do birds, of course, and for that matter, so do fish, right? And so there's all this sort of thing that we regard as convergence in some biological organisms. So most people would consider that fish are probably not strong flyers, but, even back during the Mesozoic, there were reptiles that flew, okay? So it's not just birds. And so the things that gave rise to birds, and some of them that actually evolved before birds arose. So that's called convergence, evolutionary convergence. Okay, that gets us through the background, and so let's go into fossils now. One of the things that you probably recognize is there's no way possible to find out if fossils would interbreed or whether they had viable offspring. So in fact, what we use, we call those things characters, and so characters are the way that we describe fossil taxa very commonly. And so what are the characters? And we can measure them, we can look at the ornamentation on certain fossils, and we try to make our best guess at how they're related to one another. And it's more than just a best guess, I guess. You could say that you can use statistical analyses to compare one type of organism to another and to see how they're related or not related to one another. Okay, so now back to this. So the fossilization itself. Now, you guys recognize, for instance, if I had a piece of fossil wood, that it did not grow as a silica organism. In fact, it was an organic cellular structure that gave rise to the wood, and then it was replaced molecule by molecule by molecule in the framework of that cellular structure of silica. And so the silica comes in and replaces it. We call it petrified wood. It's not really petrified in the sense that it actually has a replacement of that mineral material. So that's called permineralization is the name of that. So that's number 3 on the list here. Sometimes we have original material that's replaced in total by other minerals as well. So it's not just silica that can come in to replace something. You can have calcite, you can have pyrite and so forth. But the permineralization is more of a molecule by molecule sort of type of process, I guess you could say. For the preservation of the original shell material, that can happen sometimes, of course, especially if something is made out of calcite. It could be preserved in a limestone that way. So, for instance, brachiopods are really good at having their original material preserved. But in some cases, it actually gets recrystallized as well. So recrystallization is another process. So you can take calcite and recrystallize it, even within a limestone. So, but it's a solid state change. So that's the first 4. You could have the original material, you could have replacement, you could have permineralization, molecule by molecule. You can actually have recrystallization of that material. You can have dissolution of that material and then filling with another material. And so sometimes you can just remove the skeleton and leave the matrix that surrounds that fossil. We have names for those sorts of things as well. For instance, the animals that make their skeletons out of aragonite, very commonly aragonite is only metastable at the Earth's surface, and so therefore it goes into dissolution very, very rapidly. And so aragonite could actually be etched away within a few million years, and you can leave behind the molds or the casts that are a part of that organism. And so the cast would be the internal part, or an internal mold, if you will, and then the outer part, which is the actual mold itself. And so you would see then that's a result of the dissolution. So in, for instance, gastropods, they very commonly are made out of aragonite. Not all of them are, but some of them are. And so when you dissolve the aragonite away from a snail shell, it leaves behind a void. And so that's called a Stein Cairn. So we're going to get to that later on here. And eventually you can even carbonize, especially things that are really rich in carbon content. And so, for instance, plant debris and things like that very commonly is carbonized in the fossil record. Okay, so the next photograph shows you a replacement and permineralization of some petrified wood here. The permineralization happening piece by piece by piece, and then sometimes you can agatize that afterwards. So it's a very complex process by which you make, fossil wood sometimes. And so when the cellular structure is preserved, that's called permineralization. When it's been replaced wholesale with silica, that's called replacement. And so here's some petrified wood from the petrified forest out in northeastern Arizona here. So, for instance, in this photograph, I don't have a scale, okay? So, very important. When you're in geology, you need to have a scale in every photograph that you take. What kind of scale? Well, usually we use like rock hammers, or we use some sort of photo scale to indicate it. But you can also use something like a lens cap from a camera, or you can use even a ruler or something like that, set of keys, whatever, anything that gives you an idea of how large that is. So, for instance, when you look at that piece of petrified wood there, you don't know if that's 1 meter high or if it's just like 10 centimeters. From the size of the gravel and maybe some of the footprints around it, you might get a sense for what the scale is, however, in that photograph. And it does cast a shadow, but so do large things. They cast a shadow, but it's not a really diffuse shadow that you would see, but something that would be like in the scale of meters and so forth. So anyway, that's fossil preservation right there in petrified wood. The next photograph shows you replacement or recrystallization of some of these mollusks. These are actually bivalve mollusks right here. And so bivalves typically have a very complex shell structure to them. They have partly calcite, partly aragonite, and partly organic material. And very commonly, it's either replaced or recrystallized. Now, sometimes if it's a fairly recent preservation, you can actually get the original material. But aragonite, typically not. Not typically aragonite. So the next one is the next slide is slide 11 here. This is a Stein Cairn that I was telling you about. It's an internal cast From inside of that shell, you can see what the internal structure is made out of. So, for instance, if you look at Stein Cairns, sometimes they are, and of course that's a German word, right? So rock is Stein, and cairn is, I don't know exactly, but probably something like a cast, if you will. So here you have a gastropod on the right-hand side, and here you can see the Stein Cairn on the left-hand side here with an old wheat penny for scale there, so it's an old photograph. Okay, the other types of preservation that you might get. So there's a shark tooth. What do you think that preservation is? Well, that's probably original material. It's made out of calcium phosphate, right? So your bones and teeth very commonly are not replaced. And so that is the original material. And over on the left-hand side, that's calcium apatite, which is the same material that the shark tooth is made out of. And that's a type of brachiopod right there that's made out of, it's calcium phosphate, and it's the original material. It's the original material with some organic material. Very seldom are they recrystallized, and so that's it's a group of brachiopods they used to refer to as inarticulate brachiopods made out of calcium phosphate. Now, not all inarticulate brachiopods are made out of calcium phosphate, however. Some of them are made out of calcite as well. So there's a group called the craniids, for instance, that are made out of calcite, but they're still living today because they're not even fossil. There are fossil craniids, however. So that's an example of original hard parts there. And in fact, you see, I even listed on there for you the body fossils themselves. And then on the right-hand side, you can see a fly that's been preserved in amber. So amber, the oldest amber I think that's known, goes back to the Pennsylvanian or Permian. And so there is Permian, ancient amber, but most of it's much more young than that. And so most amber actually goes back to like Eocene age or Miocene age usually, and usually like places that have a lot of conifers that would have amber that would form, and of course flies get trapped in it and maybe even coated by it as the tree sap envelops the, in this case, the bug that was there, so the insect that was there, a flying insect in this case. So, and so the next one this is slide number 13. It's a type of replacement that's called pyritization. So not pirate, but pyritization, pyrite. And so pyrite and fool's gold, in other words. And so you can commonly get pyrite replacing some shelly organisms where they get dissolved out and then the material comes in, or you can replace it wholesale. The calcite or the aragonite, for instance, that was in that ammonite on the left-hand side, has been replaced by iron sulfide. And iron sulfide has even replaced this brachiopod that's on the right-hand side. So you're probably wondering, how am I going to be able to tell the difference between a brachiopod and a mollusk? And that all has to do with the symmetry. So if you look at the symmetry on that shell, you can see that this plane of symmetry that runs through it cuts across that shell. The left-hand side is identical to the right-hand side. That's a brachiopod. If you look at a bivalve, for example, however, you would see it has a hinge line, and the plane of symmetry goes between the two valves there. So it follows what they call the line of commissure, the opening in that valve, in between the two valves. That's the difference between a brachiopod and a bivalve. The only one that's very difficult to tell apart then are the ones that are like scallops. They have a certain bilateral symmetry, although it's not perfect from side to side as well. So next on our agenda here is some silicification. And these are trilobites that have been silicified. This is slide number 14 here. And what you're seeing here is actually kind of cool, because one of the ways that paleontologists will actually extract things like calcium phosphate from limestone, like for instance shark teeth and things like that, you can dissolve away the limestone in dilute hydrochloric acid, or even better yet, so you don't etch the fossils, you dissolve it in formic acid, or you can also take dilute acetic acid and you dissolve away the limestone, and you're left behind with some other material very commonly. And so that's going to be your calcium phosphate. And in this case, it's the silica that didn't dissolve. And so these have been pieces of lightly calcified trilobite that has been replaced by silica at some point. And so silicified trilobites, very commonly, that's a way that people prepare the trilobites, is to dissolve away the rock, and you're left behind with these sort of pieces that are replaced. And so you can still tell the characteristics of certain species that way. The heads and the tails and the genal spines and so forth of the trilobites. Okay, slide number 15, you can see some carbonization there of a fern leaf, 2 fern leaves, in fact, and so they've been compressed, and you drive off most of the volatile materials, so the water goes away, of course, and then the organic material doesn't really decay, and it just goes to carbon instead. And so you lose some of the lipids, you lose some of the resins and so forth, but you're left behind with just elemental carbon then. And so that's carbonization, a type of fossilization as well. Okay, next on the list here, this is almost a little bit of a trick here. There's a bivalve up at the very top, and it's made it with an aragonitic, partially aragonitic shell. That's that sort of nacreous layer that you see on the inside. So nacre is one of these things that will actually preserve a little tiny bit of water in there, and it gives it that sort of iridescence like that. But that is aragonite that's on the interior of that bivalve. Easy to see here that the plane of symmetry also cuts between the two valves, and so when you open it up, then they're a mirror image of one another. That's a bivalve. They used to call them pelecypods, and so that's the same thing. So bivalves and pelecypods, the same. With the aragonite in this, now this is a trick because it's not really a fossil. It's too modern to be considered a fossil. So things that have not gone through the fossilization process, we commonly call them subfossil material. If they're dead and are preserved in, let's say, sediments somewhere like a soil or or recent beach deposits and so forth, you may still get some of the original shelly material in there that preserves that sort of nacreous layer like this. And that we refer to as subfossil material. And on the right-hand side, you can see that there's a beetle husk that's essentially in some sand. And it's been partially carbonized, I would say, but some of that original shelly material is still there. Organisms that crunch when you step on them like that, like roaches, those things are made out of chitin. And so there's a high preservation potential for things that are made out of chitin. So chitin is C-H-I-T-I-N, not to be confused with what we're going to see later, which is called chitin, C-H-I-T-O-N, which is a type of mollusk as well. It's a type of polyplacophorine mollusk. So, but this chitin is a shelly material here, and that's the same sort of thing that you would find perhaps in amber. You can find chitinous insects in amber very commonly. This one's in a tar sand, however. So now tar sands are where you get natural seeps of petroleum products that escape at the Earth's surface. And sometimes if an organism was trapped in there, buried in there in some way, that the tar itself can help preserve it. And so you can imagine this because you probably heard of La Brea tar pits before. And so in La Brea tar pits, they find all sorts of organisms that preserve part of their fleshy material sometimes. Not always, but sometimes they find some of the hair and stuff like that in the fossils there. So next on the list, what type of preservation is this? I'm not quite sure. I think it's partially recrystallized because the top one up here on the right-hand side, that's a bivalve right there. It probably originally was aragonite, but it may have recrystallized. Aragonite, it doesn't always go into dissolution. Sometimes it recrystallizes to calcite. And so that was probably originally aragonitic, mostly aragonitic, and may have been recrystallized. And you can see where the shell has kind of been spalled away, that it leaves behind part of that internal cast there. And so that is a bivalve up in the upper right-hand side. In the lower left-hand side, it pretty much looks like a brachiopods, but I'm pretty sure it's probably not. I think that's a cockle shell down here, and so that probably was recrystallized as well. So when you look at preservation of these things, very commonly aragonite will be dissolved in fresh water, not so commonly in salt water. And so you have to mix, it helps you understand, I guess, the processes by which rocks form, but also the degradation of these original shelly materials. And so they indicate what type of depositional environment they may have been in. If you were to see either one of these fossils in a rock, you would say that's a marine rock. But what kind of pore chemistry, what kind of water chemistry have these things been exposed to? That whole field of study is called diagenesis. So it tells us a little bit about the chemical evolution of these shelly materials as well. Okay, so on to the next slide, which is slide 18. And this is a different type of fossil that you see here. They look like branches almost. And in a way they are, but they're branching burrows in this case. And so if you can imagine, these things would be associated with a type of shrimp, actually. So that's thalassinoides, or they could be, associated perhaps with things like Callianassa is a very common shrimp that will leave burrows behind like this. And so that's thalassinoides. It's a type of ichnospecies. And so very commonly, when we see a genus name, which is thalassinoides in this case, and you can't really identify what species, or in this case, ichnospecies it belongs to, we like to say that it is we just make the abbreviation ichnospur, SP at the end. So we say the genus name and then we say spur, which means SP period afterwards. And so spur is very commonly the use. So for instance, if we had a hominid skull, let's say, it's Homo spur, SP, if you will, and period. And it's not been identified as to what genus it would, or what species it belonged to. So that's why the ICHNSP period after thalassinoides there. Those are huge, right? So here's somebody holding and this is something I grabbed off the internet, but that's an ichnospecies here, and it's a block of sandstone that's been tilted upright, maybe by a hurricane or something like that, but it left behind these trace fossils. We call those trace fossils. They are not the actual living organism themselves, but they are the result of biological activity. And so when we find the fossil itself, we call those body fossils. And so that's the difference between trace fossils and body fossils, or species that we refer to and identify in fossil material, as opposed to ichnospecies. Very commonly, it's very difficult to identify what kind of animal may have left a certain trace. We use the best evidence we can. So, for instance, if you saw something like Rhizophycus, which is, I think, up at the very top up here, it's very commonly associated with trilobites that may have scurried across the seafloor and left behind a double track, if you will, from their little legs kicking through the sand or sediment. But that whole area of studying trace fossils, it's in a science in and of itself. It's very interesting science, mind you, because they have to rely on a lot of indirect evidence. And of course, they test their models in a more, they even build their own models to try to figure out, like actual physical models to try to figure out how could you leave behind this trace. That's the study of ichnology, or anyway, the study of trace fossils. And so they're classified by shape mostly. And so here you can see a whole selection of different types of shapes that are present here. There's difficulty sometimes determining what animal left it behind, of course. They are indicative, however, of a depositional environment. There are mostly, in the Paleozoic, of course, you're going to find mostly marine trace fossils. So if you do find a trace fossil in the Paleozoic rocks, most commonly you're going to say that's marine. But it may be marginal marine too. So by the time you get to the Permian and the Pennsylvanian, you're looking at things that actually began to live in marshy environments, for example. It's also indicative of the water quality, right? So if it's good enough for organisms to live there, that meant the water wasn't really bad water. So that's a good indicator as well. But it shows the complex sort of behavior that these animals would have had. And so that's kind of cool. So the next slide, slide 20, is a summary of the sort of animals that we're going to look at in here. Now, this is a long, long presentation, so bear with me. It's going to take a while to get through it. We're only on slide 20, and I think there's 190 out of this. So we're going to break it into parts, but we're going to go through this list essentially. So we're going to look at fossil groups. And I'm going to give you a presentation on the fossil group. So we're going to look at the simplest forms first, which would be single-celled organisms like stromatolites. Stromatolites are coccoid and filamentous cyanobacteria, or in other words, bacterium, that were photosynthetic. They could photosynthesize. They could make their energy from just absorbing the Earth's, the sun's rays, and being able to generate their own food that way. So stromatolites are some of the most important fossils because they're the first ones that kind of show up, other than a few minor little filamentous sort of things that weren't all together in a colonial sort of structure. So stromatolites are actually a funny group because they're considered to be organosedimentary rocks. So it relies not only on the organic material that was in that combined and trapped sediment, it also relies on the sediment being washed by and then trapped by these organisms. Not really organisms, it's a pre-organic or organism sort of level of arrangement in these things, but they can be fairly complex in their shapes and so forth. So stromatolites are going to be one of those groups. Forams we're going to talk about. Those are single-celled organisms. We call it foraminifera, but forams for short. Radiolaria, again, single cells, but made out of a different type of material. Diatoms as well. Dinoflagellates, both made out of silica, as are the radiolarians, in fact. Coccoliths are actually made out of a type of calcite, and that's what you see in the White Cliffs of Dover. You see it in the Paris Basin. It's the animal, or actually it's the algae that made chalk. And so coccolithospheres is what they originally are called, and then they break apart into these little plates, and those are called coccoliths or coccoliths. And then we're going to look at some of the more advanced sort of organisms, which are really not all that advanced, but we look at sponges and they have a certain specialization of some of their shell, of their cell material. And so sponges are the next level up. And then beyond that, you would have cnidarians, which would include like the jellyfishes and the corals and so forth. So cnidarians are a step above, you could say above sponges in their organization and then their cellular specialization. And so the next one is bryozoans, and brachiopods, number 8 and 9 on this list. They're both a type of lophophorate. There's a living lophophorates that are still around today called Horseshoe worms as well. So there are 3 branches of the lophophorates, and they all have a relatively simple sort of structure, although when you look at them in more and more detail, they become more and more complex that way. So as we go on here, mollusks are considered to be the next one. Mollusks probably arose from worms, as probably did the lophophorates. And we're going to break those down into the various classes. And so when we look at the mollusks, the monoplacophorans are one variety, kind of mono, implying that there's one. Polyplacophora, that's the chitin actually. And so polyplacophora, multiple plates on the backs of a mollusk. Bivalves, pretty common. You've seen those before. Cephalopods, of course, are living today. That's the nautilus shell, for instance, a chambered shell, if you will. And it's made mostly out of aragonite as well. So cephalopods are a type. I don't know why I did this, but there's A, B, C again here. So it shouldn't be that way. It should be A, B, C, D, E here. But cephalopods should be D, and bivalves should be C, and then gastropods should be E. And so gastropods, of course, they've made their way into the air-breathing environment, so they're not all like marine creatures anymore, although they were originally, they crawled out of the oceans originally. So gastropods, pretty amazing sort of creatures. They even lost their shells, and so the common garden slug is a type of gastropod that made it onto land and into your garden. Next on the list is another group that probably arose from the worms as well. That's the arthropods. Very commonly, everybody loves trilobites, so trilobites are sort of the epitome. They're kind of the first arthropods that show up in abundance on the rock record, and so trilobites and the Cambrian are like hand in hand, right? But the trilobites went extinct at the end of the Permian, so here's the trilobites, the insects, the crustaceans. Crustaceans would include things like crabs and lobsters and so forth. The next group is the ostracods, or ostracodes, and they also made it from salt water to now fresh water. And so they haven't made it to dry land yet, fortunately for us. But ostracodes, you may find them in pond water, for instance. And so ostracodes, a very specialized type of bivalved arthropod. And then there's some other ones as well. And so one of my favorite fossils, in fact, is the Eurypterid, which is the sea scorpion, which was around back during the Silurian time. They got up to be like 9 feet long, crawling across the ocean floor. And so these things were would have been probably horrifying to see one if you were diving back then or swimming in shallow water and you see a Eurypterid. It's like, oh crap! But they don't have the stinger. I think that the the insect scorpion, it's an insect. So the scorpions that we see today, the land scorpions, have a stinger at the back of the tail. But they call these, they have a resemblance, in fact, to the living scorpions, and so that's why they call them sea scorpions. That's Eurypterids. Echinoderms, that's our common version of crinoids, right? So the state fossil of Missouri is a crinoid. So we have starfishes, we have echinoids, which would be things like the diadema. It's a sea urchin, right? So sea urchins are that variety. Blastoids are now extinct. They went extinct during the end of the Mississippi, and they went extinct. So blastoids, and then crinoids, and then rhombiferans are another branch of the echinoids. And then we finally get up into things that are a little bit more sophisticated with a sort of primitive notochord, if you will. So they're called hemichordates. That would include things like the graptolites and conodonts, which we've already kind of tangentially touched on the idea of conodonts. But conodonts are a type of like grasping, like almost like teeth, if you will. They're made out of calcium phosphate again. And graptolites, of course, are made out of mostly carbonaceous material. And so graptolites and conodonts both show up. Conodonts show up in the end of the Cambrian. Graptolites show up near the beginning of the Ordovician. And then finally, we're into the fishes. And so the fishes, of course, are going to give rise to the amphibians. And the amphibians will give rise to the reptiles, and the reptiles will give rise to the mammals, and the mammals give rise to us, essentially. And there's a whole bunch of different plants that we're going to look at as well. So we're going to start with the simplest first. This part of the presentation is on stromatolites and microfossils. And that includes radiolaria, forams, dinoflagellates, diatoms. There's a handful of very small, very kind of obscure. So we think that they're relatively obscured, I suppose, because they're not, well, they're microscopic in size, some of them, so some of them are pretty small too. But stromatolites are relatively large, so we'll start with them first. They belong to the domain bacteria. They have they're eubacteria, essentially, or monarans. That is, that they are single-celled, but they don't have, I don't think in this case, a nucleus, a cell nucleus. Yeah, but they belong to the phylum Cyanobacteria. So in the old days, they used to call these blue-green algae, but they're not really related to algae. They're more closely related to bacteria. And for stromatolites, they're filamentous and coccoid bacteria. If you think of filamentous, it's easy to think a little strand of rope or a little tiny particle, I guess you could say, that's linear in form, whereas coccoid means that it's like a string of balls on a rope or something like that. So that's the sort of shape of these things. Of course, they're all microscopic as well. But the filamentous bacteria and coccoid bacteria will constitute the stromatolites. They have photosynthesis, which means that they have chloroplasts. Chloroplasts were probably separate organisms at one time, and then got incorporated into the bodies of some of these organisms that are photosynthetic. And so it's a way of making energy, essentially, if you can absorb chloroplasts. So essentially, they turn water and oxygen into sugars and then expend that energy by burning it in order to grow, metabolize, and to reproduce. So stromatolites in particular have the tendency to bind and trap sediment. So if you can imagine you're like flypaper, things will come along and stick to it at some point, not just flies, but anything that would blow onto that or get moved across that flypaper. Well, these things are sticky like flypaper, except they're in a submarine conditions, right? So these cytoplasm or whatever would latch on to sedimentary particles and layers would build up. And so what you get with cyanobacteria very commonly with these stromatolites anyway, you get crinkly laminae. So sometimes you can get just in a physical setting, you can get like flat laminae, flat planar laminae, just from the action of currents or wave actions and things like that, right? So upper plane flow, you can actually form laminae. But the crinkly laminae, you have to have something that's sticking it together. And so that's a reason why organisms, well, not really organisms, these sort of features, the cyanobacteria, these stromatolites, are more what we regard as organo-sedimentary structures. And so that's where you get the crinkling laminae like this. Now, there are some modern analogs for these things, like in Shark Bay, Australia, and then also in Eczema Sound in the Bahamas, tongue of the ocean, actually. And so they're in high energy environments or really low energy environments that are stressed due to the salt content of the waters around them. So they're marine, and they can form different shapes. The ones that are living today are mostly columnar stromatolites, but in the past, in the geologic past, they had planar stromatolites, which are relatively flat, but would have been on a surface that was like a low amplitude tidal sort of setting. And then domal stromatolites, which, as the name would suggest, they're like hemispheres. And then digitate stromatolites. So if you had a high amount of sediment being deposited over a landscape and there were stromatolites in that water column, they can maybe grow up through that sediment and then form, vertical structures essentially. And so that's what digitate stromatolites are. They're finger-shaped. So for stromatolite evolution, they're among the earliest known fossils on Earth. They're not the earliest fossil, but they are among the earliest fossils. They find them, they're about 3.5 billion years old, some of the oldest ones. That's in Australia where they found those. So stromatolites, been around a long time. They hit their peak abundance about 1.3 billion years ago. That was during the Precambrian, obviously. And they began to decline, actually, during Cambrian time. And the reason for that is probably because of the rise of grazers. The multicellular fossils began pretty much in Cambrian time. And so complex animals would come along, and of course that's a ready food source. We have to get our energy from food, and so anything that could potentially graze, and snails in particular are well adapted for eating cyanobacteria because they have a rasping tongue. It's called a radula, and so the radula can scrape off those sort of filaments and feed on them, right? So it's a ready source of grazing material, like sheep eating grass, essentially, for gastropods to come along and prey or eat and munch on the stromatolites. Now, they wouldn't have been hard. They would have been soft materials that they were eating, but that probably brought about the decline in the stromatolites at the very close of the Cambrian, or close of the Precambrian. So today, the stromatolites are predominantly in restricted marine settings, so either high current velocities or in places that have a high salinity, so hypersaline sort of situations. And they're also found in freshwater lakes, so it's kind of like slime, if you will, those sort of filaments that make up slime, if you will, in a lake or a pond and so forth. And so there are places in, like, let's say, upstate New York that have stromatolites even in their even in the lakes, so they can grow in fresh water today as well. They just have to have the water to protect them, actually, I guess, and for them to metabolize in. So the Sharks Bay, we're going to go there, and we're going to go see the ones in Eczema Sound as well. But there are marine examples even today. So the next image that you see here is Shark Bay over on the right-hand side, and that shows the location on the continent of Australia on the left-hand side. So Shark Bay is up in the northwest part of Australia. It's very hypersaline, and the reason for that is, of course, that Australia is this massively hot desert continent, and so that's the deserty part in the west, and so that's where you get this hypersaline sort of situations in the bays there. So they're in an intertidal setting there. There's a little bit of tide, but not a lot. But here you have in the next image, we're going to zoom in essentially and see where these things form. Tidal ranges can have parts that are called supertidal. That's where it is usually dry, but only during the highest of the high tides might you wet that area, and so that's supertidal. Intertidal is in between the high and low tides, and then subtidal would be below low tide, so the mean water level at low tide. So that's what those three things refer to for subtidal, intertidal, and supertidal. The closest being to dry land would be the supertidal, of course. And so there are also tidal channels associated with this as well. So the energy of the tides themselves get brought in, and it's a lot like a fluvial system in some ways. They can meander, they can do all sorts of things, but it's on a different time scale than what most fluvial channels would form. So the tidal channels you can see out here marked as A, and then B, you can actually see some of the islands that form. And at low tide, you can see land would be emergent in some of this area, and of course the supertidal setting as well. So the supertidal setting is with the 3 there. 4 is the land. Number 2 is the intertidal. That's that sort of brown ring that goes around it's the southeastern part of Shark Bay here, and then the subtidal region is in 1 there. So the next slide here shows you closer into one of these islands, and it shows you that intertidal zone there, and maybe just a hair of supertidal around the perimeter of that island. But you can kind of begin to detect little brown blotches on the seafloor. And so we're going to zoom in one more time here. And here we are at low tide, and the heads of these stromatolites are now exposed above the tide. And so you're in the intertidal zone here. And you can see how these stromatolites are growing up. And so at high tide, those things will actually be covered with seawater. And it's interesting because most of the fossil debris that's in between, it's not really fossil, most of the shelly debris that's in between these things, of course, are snails that can also live in that sort of hypersaline sort of setting. So they still prey upon stromatolites, even when they're in a setting that's just enormously hypersaline. So here's stromatolites in real life. So if you went to Western Australia, you could see these things. Here they are right where you can see right at the water line here, and you can see how they grow up in columns this way. So that's kind of a profile of what these things look like. And of course, because the shells of the gastropods who feed upon the stromatolites don't always move away real fast and they live and die, they can also be bound up because that's the sediment also that's surrounding these things. And so you actually get a lot of snails that are actually preserved in these stromatolites. So ironically, it not only feeds the snails, it also serves as their graveyard, I guess you could say as well. So those are some stromatolites for right here. We call those columnar in that shape. So they're not quite domal stromatolites. I suppose there are some domal stromatolites you can see over on the left-hand side here, but the ones that are directly in front of the lens here are columnar stromatolites here. Next slide. Now we're getting into a dominantly carbonate realm here, where you see, and in fact, most stromatolites are carbonate. Not all of them exclusively are, but most of them are. And here's an area right here. You can see Axima Sound is actually one of the deep water areas on the east side of Andros Island here, or the Great Bahama Bank, I should say. Andros Island is the dry land that's right next to tongue of the ocean. So tongue of the ocean is that sort of like dark area that's deep water essentially. And so part of that area drains off into tongue of the ocean there during the low tides. And there's not much tidal range in the Caribbean. And this is just north of the Caribbean here. But when the tides draw the water down, it actually feeds what we call oolite shoals. So there's a whole series of like sand grains that are carbonate in their composition that form along the high energy setting that's at the end of tongue of the ocean here. So the south end of the tongue of the ocean here on the southeast side of Andros Island there. So that is the sort of setting that they're in. It has normal salinity. It's a subtidal environment in this situation. There's low tidal range, but it's pretty hot down there. Although it does rain, of course, not only during the storms and during the hurricanes, but just normal rains in the afternoons and so forth. So in certain times of year. We zoom in on the south end of the tongue of the ocean here, and you can see the ooid shoals, which are these sort of sandbars that are lined up perpendicular to tongue of the ocean there, at the very south end of tongue of the ocean. And those things get moved around by the tide. So they're actually small particles that roll backwards and forwards, and they get coatings on them. And so in the modern depositional environments, they're mostly aragonitic in composition. And aragonite, of course, has the chemical formula CaCO3, just like calcite. So it's a pseudomorph of calcite, a slightly different. Well, you guys are in mineralogy. You know what a coordination number is. So it has a different coordination number than what calcite has. So here is aragonite ooids that are forming on the sea floor as the tides move them in and out. And when you get into the channels that run in between some of these, you'll actually run into some of these stromatolites. And so there's a lot of wave energy there because, well, The Great Bahama Bank is relatively flat. I mean, it gets probably no deeper than 15 feet deep in its deepest part. And so there's a mass of water that forms there, even in a very low tidal range. So a mass of water can flow in and out across that bank. And so you get a lot of wave agitation forming the ooids. And then right in between, and so next slide here, you can see some of these stromatolites poking their heads out above some of the sediment that's there. So remember, they're organosedimentary rocks. So guess what? They're going to have ooids in their composition as well. So we know that, in fact, because people have collected some of these stromatolites before, and in fact, they've sawn them open underwater. I think they're underwater saws. You can get in and cut some of the rock there. And so what they did was take essentially like a chainsaw, maybe a low tide. I don't know exactly when they did it, but I've seen photographs before where people were actually slabbing some of these stromatolites. Very high energy, of course. The diver can only get in there in between the shift, the slack water that's in between the tides. Otherwise, they wouldn't be able to maintain their sort of position in the shoals this way. So you can see the beauty of the shoals from outer space here, looking down on them. So it looks like a whole series of curtains, a lot like the Northern Lights, I guess, if you were to turn it on its side there. But that's all carbonate material, aragonite ooids, and in fact, so in between in some of these channels, you get these sort of stromatolites over here on the right-hand side. So in the ancient records, so what do the fossils look like? Some of the more interesting ones are in New York. Also, they're called cryptozoan. People didn't know what they were, right? So crypt means hidden, and some sort of creature that made them, some zoan, right? So living animal, if you will. So a zoa comes from that. And so some hidden animal was able to create these things. And so they look like cauliflowers, I guess you could say, or they have the sort of shape of a cabbage, if you will. So these are Cambrian age stromatolites from Sarasota Springs in New York, and really elegant in the way that they form here. So these are more columnar as well, but they kind of bloom and have offshoots of different branches of stromatolites in there. I'm going to show you a whole bunch of these things. So here's another one from the side view, so you can actually see the structure of these things. And so you can see each of the laminae are pretty crinkly in there. And so that's how you identify stromatolites, really. Sometimes we have to put a question mark behind them because we don't always, well, first of all, the filaments and the coccoidal sort of a structure to these things that binds that sediment together, that disappears because it's oxidized in this sort of setting and it decays. So what gets left behind are the sediments that got stuck together there. And so they get cemented, in fact. And so here you can see. Now, I suppose if you really went after it with an SEM and explore these things in great detail, you may see some of the filaments actually replaced by calcite, but that's not real common. So under an SEM, people have been able to illustrate some filaments and coccoidal cyanobacteria that way. But in fact, mostly what you see are the laminae like this when you're at a macro scale, where you can actually look at it without a hand lens or anything like that, and you can identify it, and you can kind of tell how these things kind of make their way through the sediment. They grow upward through the sediment. On the next slide, you can actually see the top surface of a stromatolite, and here you can see the columnar sort of shape to it, and there may be some burrows in this as well, but in the right-hand side, you can actually see the columns as well. These things form reefs, so you can see that they grew off of a relatively hard substrate that is a limestone bed that's below them. And then they grew upward, and then they would have flank beds off to the side. And so the flank beds would be on the left-hand side in the upper right, and then the lower right, you'll see the flank beds off on the right-hand side. Actually, both sides. And so you get columnar stromatolites here, and a little bit of interstitial material that will fill in between the stromatolites also. The reason that these things are preserved partially is because of the flank beds that are around them, and then also because of the shale that blanketed over the top then of those flank beds and the stromatolites. And so the one on the lower right is from the Shell Creek Mountains in the eastern Nevada. The one at the top, I think, I don't recall exactly where that is. It's probably from western Utah, I'm guessing there. Certainly one from western Utah on the left-hand side, and that's from a rock unit called the Hellemeria Dolomite, or the Hellemeria Formation. Next slide, we have some from Texas, and these grow up again. You can see that they have that columnar shape again. There are recursive patterns in geologic history, and one of those recursive patterns is that reefs sometimes maintain the same shape through time. And so this is actually a spur and groove sort of setting that these things were deposited in. You don't see the grooves on the sides of this, but these would have been kind of upright and growing vertically, and then around these things there would be tidal channels perhaps. And you see the same sort of thing in modern coral reefs, and so the coral reefs have that same tendency to form spur and groove. The next slide, here's some more that show the top surface, and in fact some of these things can actually be burrowed. So you get kind of a core that's been burrowed, burrowing worms and all sorts of other organisms that are looking for food in the stratigraphic, in the sediments that may be surrounding them. They may land on a spot where it's like, oh, this is delicious, and they'll go after it and burrow into what in this case would probably be the center part of a stromatolite. And so sometimes stromatolites aren't perfect, in other words, and so they may have burrows incorporated into the middle parts of them, and then the laminae will be on the perimeter, of course, where these things are growing upward. And then also, it brings to mind that there are other types of organisms other than just stromatolites that are known as thrombolites, and thrombolites would be things that we don't have real good evidence for the layering in them, but we do have evidence that they're microbial from the irregular shape and the fact that they may form reef or certainly resistant sort of structures perhaps in the sedimentary record. So here we have some from the top. The next slide you can see some more from the top. Here again, good laminae around the outside and maybe some burrowed fabrics on the interior of that. Next slide, you can see the same sort of structure. In fact, I think they're the same stromatolites in the upper part of the Animalia formation here. So these things are kind of like loaves of bread, if you will, as in top view, but in vertical view, you would see that these things are probably about 1 meter or so in height off of what would have been the seafloor at the time. And they would have probably grown and kept pace, and so maybe pace with sea level rise. So in this case, they probably were no more than just a few centimeters above sea level above the sea floor at any time, because the sediments would be piled up around them essentially. And so they had to keep in a position where they could still photosynthesize. So they had to be exposed to the sunlight essentially in the shallow water column. Next slide, you can see some more of these same sorts of stromatolites. And I know for a fact that these actually come from the Animalia formation here. And so these have that burrow fabric in the interiors and then the layered of the laminae around the outside. Then, and stromatolites are absolutely gorgeous. In the next slide, you can see one in cross section again. Here, very faint, of course, but you can see the laminae that stretch across there. And then there's a surface of unconformity at the very top up there. It's a carbonate on carbonate unconformity, kind of disconformable surface, a very short sort of diastem in the geologic record, a very short period of time that may be missing there, but rocks have been removed by erosion at that surface where you get the slightly darker carbonate at the top than what it is in the rest of the rock there. So if stromatolites are right at sea level, it doesn't take much of a sea level fall to expose them to the rigors of what we call diagenesis. Next slide are some absolutely gorgeous columnar stromatolites, and you can see them growing upward here with flank beds in between that are slightly burrowed perhaps as well. And so that sort of orangish material that you see interstitial between the actual stromatolites themselves, that material in fact would be probably slightly dolomitic as well. So now we continue on with some of the other single-celled organisms that we need to talk about, and that would include the foraminifera and the radiolaria, the diatoms, the dinoflagellates, and there's a handful of other ones as well. But for now, let's talk about the microfossils, the radiolaria first. So they belong to Protists. Protists are single-celled organisms, of course. They do have a cell nucleus, places them in the eukaryotes. And so radiolaria have been around a long time. The most interesting thing about them, the most important thing, of course, is that they are very, very tiny, ranging from 30 microns up to 2 millimeters in size, and they make their skeletons out of silica. So that silica typically is a lot like opal, if you will, and so that silica forms rocks like novaculite. So novaculite, if you're familiar with the Arkansas novaculite, you're ahead of the game here. So they are small and spherical in shape, so let's go ahead and take a look at them. So that's on to the next slide here. And you can see that they have very complex shapes, and so most of these photographs on the left-hand side were taken with either an SEM or under high magnification with a petrographic scope, and so they separated them out obviously here, but they're like small bell shapes and small circles, small spheres, and so forth. And very elaborate sort of forms, and of course they're perforate as well, so they can exchange their protoplasm in the with the water column, get their nutrients out of the water column. On the right-hand side, you see an illustration by I think it was Ernst Haeckel, who studied many of these things early on, and if it's not his, it's somebody who took after him. Ernst Haeckel is a very famous biologist who did a lot of elaborate etchings of these sort of features, and so these creatures. On to the next slide, you can see one of the simple ones here that has a sort of a spherical sort of shape to it, and it's a sphere and a sphere for one of them, little spines on them and so forth. But again, they're made out of opal. Opal is a type of silica, and eventually it can become more like quartz at some point, and so that's what novaculite is. It's usually, most people regard it as what they call radiolarian ooze. Now, in the deep oceans, you're more likely to dissolve all the carbonate material down there, but the silica remains, and so you can get this ooze on the ancient seafloor. Is what they think, and I think it's even in the present day too, because radiolarians, of course, are still with us. They're from early Cambrian all the way to recent. Next on the list, we have the foraminifera. Again, single-cell organisms. They have a cell nucleus, so they're a eukaryote, and so they also are marine and non-marine, and of course, they're living today as well. This is what small animals that filter feed would eat out of the water column. And so forams in general, that is, but they do have shells on them. And so there's a couple specific varieties, in fact, and some of them are actually carbonate. And so some of them are made out of calcite, essentially. And so when they're made out of calcite, of course, they can form fossils. And so there's some that are really important in the ancient record, and of course most of these are actually pretty important in the modern record as well. They live at certain water depths, and so some of them are planktonic, but many of them live in the ocean floor, on the ocean floor, and those would be called benthic. Okay, so that's the difference. The planktonic would be the swimmers, and the benthic would be the ones living on the ocean floor. Benthic forams have been very useful for describing climate change in the recent geologic past. And so we look at the Delta 18O record of these sort of creatures, and there seems to be a correlation then with the times of global ice ages and so forth. And so forams pretty important for that sort of work. For the Cambrian to recent, they're a very diverse group. They have coiling ratios, in fact, so some of them go left-handed and some of them go right-handed, or clockwise or anti-clockwise. And so that's related to temperature, apparently. And so temperature is a very important aspect for where these things live. The oil industry loves people who work with forams. And so they work with modern forams mostly, and so you can tell exactly what age of rock you're in from these forams. And so in the geologic past, there are different forms that lived, and of course the paleontologists would then be able to tell the oil company executives exactly, it's like, okay, these are this age and they are, related to this bathyal depths. And so it helps with the interpretation of the depositional environment as well. So they go from 100 microns, slightly larger, of course, than radiolaria, up to 20 centimeters in length. Now, 20 centimeters for a single-celled organism. Think about that for a minute. I've seen some really big ones myself, like about 4 or 5 centimeters long. They are particularly large in the Permian, in fact. And so in the Pennsylvanian and Permian, they were what we call index fossils. Index fossils tend to have a short range, they evolve very rapidly, and then they have a widespread distribution. The group of forams that existed back in the Permian and actually before that in the Pennsylvanian were called fusulinids. And so fusulinid biostratigraphy was really important. In fact, I've got a couple of friends who studied those things from back in that time period. And So pretty interesting rock. So fusulinids are an index fossil. Index fossils, short taxon range, and then they have a a rapid evolution, and then, of course, they're widespread. This next slide shows you what they kind of look like here. These are modern forams, of course, and there's some radiolaria pieces in there as well. The forams are more globular in shape, and so the bits and pieces that look like sieves are more likely the radiolaria, as you see here, identified in this image. And so these are ones that are roughly about the same size here. If they're not made out of calcite, they are made out of agglutinated material. Now, what agglutinated means is it's been stuck together, essentially sand grains that have been applied together. And so there's a whole group of forams who make their skeletons that way. On to the next slide, you can see another foram here with a very elaborate, slightly perforated sort of, these things are called tests, actually. So a test is the skeletal material, external skeleton in this case, and so it's slightly perforate so they can have interchange, with the outside seawater in this case, but kind of a spiral shape, in fact, here. So you can see that they do coil commonly. So that's one that's made out of calcite right there. On the next slide, you can see some that are agglutinated, but also just different shapes. So biseriate, spiral, planospiral sort of organisms as well. They're existent, for instance, in Florida. If you dive in Florida and pick up a handful of carbonate sand, you're going to see a bunch of forams in it. So some of them have that sort of football shape you can see here as well. That's in the upper right-hand side, and that's sort of a coiling as well, coil like a spiral that's kind of like a tip-to-tip sort of spiral. But that's what the fusilinid sort of shape is like as well. In the next slide, you can see an agglutinated foram on the right-hand side. On the left-hand side, you see some that are calcareous, and they are made out of calcite, of course, and spiral in their shape. The next slide, here you go. Here's a fusilinid right here. Here's one that has that sort of spiral shape, but it's, again, tip-to-tip. That it's spiral, so it's shaped like a football sort of, and so the spiral goes around like you throw a football in that sort of spiral shape, and you can see the interior cross-section on the upper right here. And they call them wheat fossils, in fact, and so that's the state fossil of Kansas is Triticales. It's named after triticale wheat. So they look like little tiny wheat grains in the carbonate rock that you see here. Penny for scale there. Next on the list, and so we're still in the single-celled organisms here. This is a chromista here, so this is going to be one that is regarded as a plant, okay? So it's photosynthetic, and they're marine and non-marine today, and so diatoms can occur in lakes or they can occur in the ocean. They're extremely abundant. Diatoms, you may have heard of diatomaceous earth before. Well, those are the skeletons of these little creatures that would have existed at one time. And were now preserved as a sediment, perhaps, but they are living in the modern oceans and in some fresh water as well. Unicellular, or sometimes they can be colonial. They also have a siliceous skeleton called a frustule, and these are really tiny things. And even radiolaria would eat these things for their meals. Okay, so radiolaria would munch on things like diatoms. So extremely small sort of creatures right here. They're spherical, cylindrical. They have a little bit more complex shape, I guess you could say, than what the radiolaria have. You may know another group of organisms as well. They're called dinoflagellates, and they also are associated with photosynthesis, but also they are plant material essentially, but they can live in corals, and in fact they have a means of locomotion as well. So they're not entirely photosynthetic. Some of them are responsible for what they call the red tide in places off the coast of Florida, for instance. That's the thing that kills a lot of fish commonly. So they have blooms essentially in some of the shallow ocean waters around Florida, killing the fish. And so that's the red tide. Here you can see on the next slide some diatoms. Don't really have a scale on this, but they're made out of silica again, as are the dinoflagellates. So the next slide shows you some more diatoms here, and then you can see more diatoms in the next slide and more diatoms in the next slide. And so just odd things that are very, very tiny, single-celled organisms. And then lastly, we're at another one that's a chromista here. This is the coccoliths, and so coccoliths are actually made out of carbonate material. They're responsible for what is chalk. The rock chalk is made out of coccoliths plates. So, for instance, the White Cliffs of Dover are going to be made out of these little tiny organisms that would have lived in the water column, died, and their plates fell apart and fell to the ocean floor. Not deep enough to where they would actually get dissolved in the cold, deep waters of the oceans, however, shallower sort of settings somewhat. So even in Kansas, even in central Kansas, they have what they call the chalk hills there, again, made out of coccoliths. They're very small. These plates are very small. It takes an SEM to be able to image them. You can see that in the next image here. So here's a coccolith. That is a coccolithosphere. And then here's the coccolith plate on the right-hand side, on the side. So you can see how the plates go together to make these sort of like little algal balls, if you will. Here's a coccolith on the next slide that look like trumpets, essentially. And so they fall apart as well once they die and will become part of the sediment that's on the sea floor, again, forming chalk. And so that is the extent of what we're talking about with some of the simplest organisms on Earth. And so it's 11 o'clock right now. I got to get in. But anyway, I will talk to you soon, and we'll start again with the more multicellular organisms. We're gonna drift away from the single-celled organisms and get into these sponges, which are the simplest variety of multicellular life on this planet. So that's it for now. Thanks now. Bye. So on to sponges. Sponges are very interesting sort of animals. They are an animal, not a plant. They can have some photosynthetic algae in them as well, however. They do have a specialized cell organization, so they can have 3 or more different kinds of specialized cells within them. They're eukaryotes, obviously. We're going to deal with eukaryotes from here on pretty much until we get to plants. They're an animal. They're called Phylum Periphera, so periferin is the other word. People have recognized these in Ediacaran, possibly. They're very difficult to know what you're looking at in any Ediacaran sort of faunas exactly. But they have that sort of like capacity to have an opening. And so that opening at the top is called an osculum, and they have what they call an atrium inside of that. And so the atrium allows an exchange for water laden with food particles, perhaps. And so plankton and other sorts of organisms that may float by, sponges will try to filter it out. And so they're filter feeders. And so they have another ingredient in their fossils as well, so they can actually secrete a skeleton. They can be calcareous. They can be made out of silica as well. But sometimes, and you may be familiar with this, they're made out of spongin. So spongin is a sort of organic material. And so sponges are very absorbent, as you well know. But that's just the spongin part of the organism itself. It doesn't include the soft parts or the organic materials that are not spongin that would have been left out to dry. Let's say if you're a sponge diver in the Mediterranean, in Greece, or in other places. They even do it in Florida. And so you can dive, grab a sponge off the seafloor, and then what they do is they dry them out, of course, and then dispose of all the organic materials that are in there. Mesenchyme is the material that tends to be around the spongin, and so that's kind of the middle part of the organism itself. The spicules can be hard parts that give it a structure and a rigidity, if you will, and they can, as we said already, calcareous or siliceous here. The atrium is the internal part, the osculum is the opening, and then they also can feed based on Bernoulli's effect. And so from physics, you're probably familiar with Bernoulli. You can blow across the top of a cylinder and you make a noise. You have a reed in there, you can make some sound out of it. You have this idea that if currents flow across the top, across the osculum, it will draw water into the atrium through the organism itself and allow it to filter feed that way. So there may be a lot of different organisms that got clustered into and included in with the sponges. There is an ancient group that was among the first living organisms that are recorded in the Cambrian, and so these were going from lower Cambrian into the lower middle Cambrian, and they're called Archaeocyaths. Very elaborate sort of creatures, wagon wheel sort of shape and cross section, but like little tiny vases that would have an opening at the top and then an atrium that wasn't nearly as closed off as what you see in this diagram. Those are the Archaeocyaths, but lower to lower middle Cambrian in the rocks, of course, now we're talking about. And then there's sclerosponges, ones that are like Stroma Toperoids. And so Stroma Toperoids formed reefs back during the Silurian, and actually even in the late Ordovician as well. And they existed all the way into the Cretaceous here, but they tend to be more binding sort of agents, I guess, on the seafloor. But they're not with us anymore, so we don't have living a group of organisms that would be assigned to the sponges called the Stroma Toperoids, not to be confused with stromatolites. So Stroma Toperoids are a type of sponge, if you will. They used to regard them as a type of enigmata. That is, nobody knew exactly what their affiliation is because they didn't have a really well-developed atrium or anything like that. But when people study these things, it's like, well, they have all the other characteristics of a sponge. Let's put them in with the sponges. Finally, there's one last group, the Sphinctezone Sponges, which are kind of like chains of balls, essentially, that are lined up, but they all have atria. And people think that these are the descendants, in fact, of some of the irregular Archaeocyaths that existed all the way back in Cambrian time. And so then they're regarded then as a type of offshoot, but of course there's no living ones today, so we don't get to study them in any great detail. So I'm going to show you some of the history first. Here's essentially the tree of how these things evolved. You can see the Sphinctezoans all the way over there on the right-hand side, and Stroma Toperoids in the middle there, and then the Archaeocyaths are at the very bottom. There are just a handful of actually, I was in the publication, and I found some of these Middle Cambrian ones, Upper Middle Cambrian Archaeocyathids. And there are some ones that have been argued to be in the Upper Cambrian as well. So, but Upper Middle Cambrian, Upper Cambrian, question mark, I guess you could put on those. But the calcareous sponges, they're still around with us today and living on the seafloor. So a pretty diverse group, even though they're very simple organisms. Here's one in a photograph of one in the next slide. This is slide number 64 in the presentation here. And you can see that it looks like a whole series of vases that are kind of clustered together. In the next slide, you can see one that's covered by all sorts of sediment and surrounded by turtle grass that's encrusted with forams and other sorts of little tiny organisms, if you will, but that's turtle grass surrounding a sponge there. So that's a sponge that would have access to ocean water and be able to filter feed and make its metabolize ingredients out of the water column that way. So they filter feed. Next slide. You can't really see it in this photo, but the next slide will show you in detail what these things are. This is from Miami. This is one of the canals that runs through South Miami, and here you can see actually a whole series of sponges that would have been exposed in sort of a back reef setting here. The next slide, you can actually see the interior of one of those. So they can get to be fairly large in the openings for these things. And so this would be one that is Pleistocene in age. Now it's covered with modern-day sediment, and of course they dredged the canal through and exposed it. In the next slide, you can see some of the varieties of sponge spicules. Most of these look like they are siliceous sponge spicules here, and that's a very common sort of like material that by which they would make their spongy material, their spicules out of. Spicules give them the sort of structure that they have. I've seen some that form exact mats, in fact, in the Cambrian, where they've been, it looks like it's been rolled out. But a sponge that has been, in other words, opened up from its atrium and then exposing all of the remaining bits and pieces that didn't decay. In the next slide, you can see an Archaeocyath. That's what we regard as a regular Archaeocyath. They have a sort of porous network on the outside, but they have two layers. There's a layer on the outside and a layer on the inside, and then there's an intervallum that's in between. And so the inner cone and then the outer cone, and then they would root themselves down on the sea floor. In Antarctica, I found one of these things. It was 97 centimeters long. They would essentially have their heads exposed above the sediment interface, but it was no more than the size of a pencil at one end and maybe the size of a silver dollar at the other end, and 97 centimeters long, exposed in what today is a carbonate rock. And so kind of neat. Regular Archaeocyaths here is what you see. On the next slide, you can actually see a Stroma Toperoid. There are similarities between the structure of a Stroma Toperoid and an irregular Archaeocyath, but these are Stroma Toperoids, and they form like little mats, if you will, and they have little pore spaces at the top. A mammalose structure, they call it, at the very top up there. They're sort of rounded knobs, if you will, and so that would allow the circulation of water to penetrate into these sort of planar sort of features. So these are planar Stroma Toperoids here, and so they're calcareous, of course, and they go all the way from Middle Ordovician all the way into the Cretaceous then, and the later ones are in deep water mostly. Stromatoperoids important. Some of them even are like what they call spaghetti stromatoperoids. I've seen some of those in the Devonian out in Utah and Nevada. Amazing sort of diversity within these forms themselves. And so, again, these things are a type of sponge, and so they are sponge organisms that are animals. Now, there are some things that look like sponges and have been interpreted in the geologic past as sponges, but they probably aren't. They would have been like clusters of algae around a core, if you will, like an empty vessel, essentially, and they get flattened through time as well. And so that's one over here on the lower right-hand side. This is on slide 71 here. Now, that's regarded as a problematicum, although most people regard these now as a type of dasyclad algae. So dasyclad algae are probably related to the calathid organisms that formed reefs also back during the lower, actually in the middle Ordovician roughly. But these receptaculidids have a sort of sunflower shape to them. If you look at a sunflower head and you look at the arrangement of seeds in it, it follows what they call the the Fibonacci series. Yeah, so if you add 1 and 1, it equals 2. Add 2 and 3 together, it equals 5. 5 and 3, of course, equals 8. 8 and 5 equals 13. That's the Fibonacci series, and it gives you that sort of like complex spiral, like a spirograph. Back when I was a kid, we used to have toys like that, where you could make these spiral sort of shapes like that. That is the Fibonacci series. However, how these things expand outward, and so receptaculidids follow that pattern, as do sunflowers. And you'll find the Fibonacci is one of those easy sort of progressions to understand mathematically. So a lot of people use that when they make television shows to try to impress people by how smart they are or something. I don't know. But anyway, that's a receptaculidid on the right-hand side here. Calathids were probably related to them. They look a little bit like, there's a fine line, I guess you could say. These are animals. They may have had photosynthetic algae within them as well. But it's hard to know when, most of that soft material is now gone. We're just left behind with the fossil itself. Okay, so next on the list here, we have Cnidaria. So we're getting into slightly more complex organisms. These are things that will like sting you, okay? So that's a good way to remember them. Jellyfish, of course, have the nematophores, which would allow them to, nematophore cells, specialized cells that will actually paralyze fish when they come in contact, and so they'll actually eat fish that are stunned by these cells, essentially, and they'll also sting human beings as well. In fact, box jellyfish can actually kill you. The ones from Australia, right? So you have to be careful with some of these things. Things like the Portuguese man of war are slightly related to this. They're a siphonophore. They are a type of cnidarian. But they're not on the list of 3 here, but they would be included in the others there. So jellyfish, corals, and then siphonophores would be another one here. It used to be that cnidarians were actually regarded as coelenterids, and the coelom is the center part or the hollow part of that organism, and so there would be specialized cells around that coelom then. I'm not going to get too deep into the classification here, but you can see down here at the bottom, there's scyphozoans, anthozoans, which would include the corals and the anemones, and sea pins are another variety of anthozoan. Cubozoids are the ones that have the box jellies, the ones that will kill you in Australia, but they have no known geologic record that I know of anyway, although they find new things all the time, okay, and people look for these things. And so unless you're a specialist, you may not know that. Hydrozoa is another one, fire coral. Siphonophores that we talked about, like the Portuguese man of war. There's some other siphonophores out there as well, like Propita and Velvala, by the wind sailors. And then there are hydroids that will live on the seafloor. They're benthic in their life life mode, if you will. These things tend to have two different generations, I guess you could say. Part of the time that they are medusoids, they will form through sexual reproduction as medusoids, and then they will finally form a polyp. And so they have both sexual, the polyps have the asexual variety, and so they just bud off and release these medusoids, but it's the medusoids who have sexual reproduction then. And so they have tentacles as well, right? So that will trail behind and allow them to kill something essentially and draw it up, and so they can pull it up into their mouth and then digest it through pretty much osmosis. They have no true anus or anything like that, so the very primitive sort of like digestion that they have. In the next slide, you can see here's some varieties of these. There's a hydroid, hydrozoan at the bottom. Here, like a fire coral that lives on the sea floor. Scyphozoans, of course, that you can see here in the cubozoans, and then the anthozoans are the true corals there. The next slide, it just tells you what a scyphozoan is. That's a jellyfish for the most part here. They think that they find these things in the Ediacaran as well. It's one of the more common ones that you would find in the Ediacaran, perhaps. Now, the Ediacaran was actually defined from the Flinders Range in Australia, and it is a place in south-central Australia, a very deserty sort of setting, where you get these organisms preserved in Precambrian rocks, right? And so it was a guy named Reg Sprigg who actually found these things back in the 1940s, and they were described in the late 1940s and into the 50s as being, oh, hey, here's something that's even older than the Cambrian rocks that we recognize, and there are fossils in them. And so Reg Sprigg went out to find those, and he did. They find these things in soft body preservation in places like the Margin Formation in west central Utah. They find them in the Upper Cambrian in various places. They find them stranded on beaches in places like Wisconsin, and so some of the Cambrian in Wisconsin has some of these things preserved as well. Here's some of that life form here. You can see on the next slide. This is slide number 76 now. You're looking at the jellyfish life cycle there from the medusoid to the budding polyp at the bottom, and so the buds snap off and then form these medusoids. And then they reproduce sexually, and that forms the polyps then, which land on the benthic substrate of some sort. And so they'll spend part of their life cycle there. You can see the enteron, or the coelom, as you would call it, the gut part. On the left-hand side here, there's the mouth at the very bottom, and then, of course, the tentacles would hang off the bottom there as well. So these things are really pretty spectacular. If you look in the next slide, here's an image of one of the ones from the Ediacaran from the Flinders Range in Australia. This thing's called Dickinsonia, named after Dickinson. And so this is a type of cnidarian here. Most people would agree. Very simple sort of body plan, but flat and preserved essentially as an impression in sandstone. In the next slide, you see some more of the fossil materials here. Only one of them has a little bit of organic material that may still be present in it, and these are all Cambrian jellyfishes here. You can see the ripples around the one on the upper left-hand side, and actually the lower left-hand side as well. But there's some carbonaceous material in the middle section there, and that shows you what a recent one looks like on the right-hand side there. So corals, on the other hand, are much more interesting than the jellyfishes, which is nice. They have an external skeleton made out of calcite or aragonite, and so they've changed through time. The living ones, most of the living ones are actually made out of aragonite these days, and they belong to a group called the, here they are, Scleractinian, down at the very bottom down here practically. So the Scleractinians, in fact, have zooxanthellae in them, and so they have the capacity to take in algae and the dinoflagellates and things like that into their cellular structure, where these things are then protected from being, absorbed as a food particle, and they will actually photosynthesize, producing sugars then, of course, which the coral is happy to devour. And so it's a very, what they call a symbiotic or a commensal relationship. It's least symbiotic. Commensal is a little bit more in favor of one animal as opposed to the other, and I suppose the corals are a little bit better off than their zooxanthellae in some ways. But anyway, so the corals are the anthozoans, okay? So there's a lot of different varieties back in the ancient record. There are tabular corals. There are also corals that were solitary. There are corals that were colonial, and they have formed reefs through various times in geologic time, all the way back, for instance, in the Triassic, they were reef formers. And of course, they form reefs today, like the Great Barrier Reef in Australia is predominantly coral. So of the corals, there's a whole bunch of them, and they rank them basically based on their appendages and what kind of body parts they have. And but in fact, in the ancient ones, in the fossil record, we're usually interested in whether it's tabulate or a rugose coral. And then the Scleractinian corals don't tend to be preserved quite so easily in the geologic record because there's not a great record of aragonite in the geologic past. So, but the rugose corals are often called horn corals in many cases because they're solitary and they have sort of a structure that looks a little bit like those Archaeocyath that you've already seen here. So you should be able to recognize the difference between a rugose coral and an Archaeocyath. So that's going to be one of the probable questions that you'll see on a quiz. That's up to Jared how he wants to quiz you on these things. But yeah, anyway, on the next slide you can see one of the tabular corals. They call them tabular corals because they're more or less flat and they will encrust things like this. So that's Fabicides right here. Some of those are in the recent record in the Mississippian around here. Syringopora is a very common Mississippian coral as well. And here you can see like the tubular structure to the Syringoporas, and but in fact they have little tables in their structure that would tell you that it's a tabular coral. If you go back to the Fabicides, you can see you're looking actually at the bottoms of some of the corals where the corallium would actually exist inside of that sort of space that's on the coral. So the tabular corals very commonly have small coralia associated with them. Halicides is another one here, slide number 82. Again, you're looking at a coral there. It's you could call it a chain coral as well. In the next slide, this is actually the state rock of Michigan, and it's called the Petoskey Stone. But it's a type of rugose coral, which has these septi, these dividers in there. You can kind of see them kind of radially shaped around the edge of that corallium. And then there are dividers, the cell wall. It's the actual corallium itself is defined by these walls that are around it. And so this would have been a colonial rugose coral, which is kind of unusual. Hexagonaria here. And they, in fact, call that the state fossil of the Petoskey Stone, the state fossil of Michigan, because they find it up around Traverse City in that area. And so on the next slide, you can see a solitary rugose coral. Rugose means that it's rugged or wrinkled. And so you can see how they have the various life stages in the middle part here that shows you the upward growth of these things. They would grow in the sediment a lot like an Archaeocyath would. You can see the corallium is at the very end of it there, and that's where this thing that looks like an upside-down jellyfish lives. And of course, corals have the capacity to sting people as well, or fish for that matter. And so instead of stinging small fish, in fact, some small fish get away with it, like the clownfish that go in and clean out the debris and the detritus in these things. But you can see a rugose coral on the right-hand side there, and that's the structure on the left-hand side. In the next slide, you can actually see a sea anemone. And so sea anemones are a type of anthozoan, and they have this unusual capacity to be able to move a little bit, right? So they're not necessarily just attached to the seafloor. So people have suggested that they can actually crawl through the sediment, and so they can leave behind a trace fossil called Conictus. So it looks like a whole series of cones, essentially, where these things have escaped from being buried in sediment, essentially. You can see some conictus on the next slide here. Is the trace fossil of a sea anemone? There's still some question about it, so I can't tell you definitively if that's the case, but that is the Miami oolite that's next to it there, and that's Pleistocene in age. So we got to see that on one of the field trips that we did out of Missouri State here down to the Florida Keys, and went through Miami. And here you can see a Scleractinian coral. Now, it's not really well preserved because it's beginning to be recrystallized now. As it turns out, aragonite can be dissolved fairly easily. So you can see, in fact, in the Miami Formation here, there's a Pleistocene Scleractinian coral. The next slide shows you another one that is still well preserved because it's been quarried out somewhat. And this is in the Key Largo limestone, a little bit farther south in a place called Winley Key. As you're getting into the Keys, past Key Largo, the Key Largo limestone crops out in some of the higher keys. And so they're Pleistocene in age. They would have been deposited there back, during the last interglacial period, roughly 125,000 to 80,000 years ago, when sea level was higher. These corals actually grew on those islands. And then as sea level dropped, of course, it exposed them, and they became what you see here today. Just what's barely considered to be fossil material, and that's a Scleractinian coral. They're a big one, mind you, so that's like a brain coral right there. And a bunch more of these. So I have several of these photographs. We stopped at Winley Key on this field trip and looked at modern carbonate depositional environments here. So there's another Scleractinian coral there. In the next slide, you can actually see it on the sea floor at Taverner Key, where we did a short dive there. And these are finger corals, a type that's called Porides. And so Porides here growing in amongst the turtle grass here. So Thalassia is the turtle grass there. That's the greenish parts that you see. And of course, the corals themselves look like they're dead, but in fact, they may be dead, but they may still have some Coralia that are still up and living. And so these things get washed around and hit by hurricanes all the time, and sort of they can be devastated actually, but again, they can make their way back. In the next slide, you can see what looks like a Monastria. It's a Scleractinian brain coral from Florida. This is Samba Key, I think it is, off of Key West. Here's another slide. The next slide, it's a little bit murky water the day that we were there. If you want to see one of my recent photos of this, there's some from Jamaica. One of them was in the GSA calendar last year. No, actually for 2020. I think it was July. But anyway, I've got a slide that's going to be published next year with the 2021 calendar as well. I'm pretty happy about that. Here's some Scleractinian corals anyway from Florida. You can also see some soft corals in there as well. The pulpy sort of ones that they don't have the hard skeletal structure, obviously. And slide number 93 here. This is an Octa Coralia. You may have seen like coral jewelry where they use coral and turquoise to make rings and things like that. So the blue and the red together in sterling silver usually. This is the coral that they use for that, and it's a type of Octa Coralia here. So it's used for jewelry mostly, I guess you could say. So it has that red color. And it's calcareous in its composition as well. Okay, so that takes us through the Coelenterids, or the Cnidarians as they're now called. So that is one of the more simple groups, a little bit more advanced, in fact, above the sponges. Next, we're going to get into the Lophophorates, which would include Furonid worms, which are living worms today, and then the Bryozoans, which were also living. They're in marine waters as well as fresh water. And then the brachiopods, which are fully marine, but they still live today. And so they go all the way back, in fact. The bryozoans go back to Middle Ordovician time, and then the brachiopods go all the way back to Lower Cambrian time. And the Furonid worms, we don't know, because they don't have a great fossil record, because they don't leave much behind for hard parts. So in the next slide, you can actually see what a lophophora is, and that's what links all of these groups together. The lophophora is a feeding structure that allows them to suck the goodies out of the water, if you will. So they'll eat things like radiolaria and other sorts of microbes, if you will, that are floating, planktic microbes. And so it's kind of a feathery sort of structure. And so the lophophore is common to the bryozoans, it's common to the brachiopods, and also the Furonid worms, and that shows this linkage. And so that's what unites the Lophophorates. And so the Lophophorates include the Furonids again, and then also brachiopods and bryozoans. And so here at the bottom on this these two photographs, you can see the Lophophora in all of its splendor from a Furonid worm. So Furonid worms are kind of like tube worms, if you will, and horseshoe worms is the acronym the nickname they go by. And they have a chitinous skeleton, but they're not going to be found very commonly in the rock record. Usually, if they are described, they're going to have a question mark after that recognition. And so So Phoronid worms may go back all the way into the lower Cambrian, but you know, it's like you're making guesses when you do that sort of thing. Hopefully you'd see a lophophore though, and maybe preserved as a soft part preservation and something like that. The next group we're going to go on to is the bryozoans. The bryozoans are actually fairly complex sort of organisms, and so you can see they have like on the right-hand side here, you can see their lophophore is a little bit more simple than what the phoronid worms are, but they have a sort of a head region there, if you will, and I think they have a light-sensitive organ that's not exactly an eye, but they poke out of their little living space, if you will, and grab some plankton that may be floating by, and then they withdraw into their living space again. And so those things are called zoecia, as I recall. And typically when you see these things in Paleozoic age rocks, they're going to look and have certain forms to them. And so that's where we just come up with the easy forms to classify them as twiggy, mounded, encrusting, lacy, and other. They're going to look a little bit like some of the tabular corals, but the corals are going to have larger coralia, and so the space for living, you know. So the bryozoans themselves have fairly small sort of space to live in. The fenestrate bryozoans are the ones that form like a meshwork or a framework, but the twiggy ones are more like on a stalk. They look a little bit more coral-like, I guess you could say. You might confuse them, I suppose, with something like Porides, but typically you're not gonna be able to have any difficulty, I don't think, differentiating them from corals. They make their skeletons out of calcite, and so that's another thing that they have in common with corals. But so the next slide gives us an idea of what some of the lacy bryozoans look like, and these are the fenestral bryozoans. Some recently living material up there on the top, and then you can see some of the fossil material down here. And again, lacy is a pretty apt description for these sort of creatures. The creature itself would live in the crossbars, essentially, between the openings on that, and so the openings would provide for currents to flow through and deliver some of the plankton and so forth right in front of the lophophores that would be extending out and filter feeding. In the next slide, you can see some of the apparatus that would hold up some of these lacy sort of materials. I think you saw some of this in the field trip across the campus where you saw Archimedes. Archimedes is a type of fenestral bryozoan, or a lacy bryozoan, if you will, but it's the central core that held the whole thing together. It's kind of like a spiral that would have wrapped around, like a curtain-like spiral. There's a little brachiopod actually on the fossil sample up at the upper part there, and of course that is brachiopods, and the bryozoans, of course, are related to one another. That's an individual as opposed to a colonial sort of lophophorate there. Now, on the next slide, you can see some more of the twiggy bryozoans here. So twiggy bryozoans would have that sort of aspect, and maybe some of the lacy ones over on the left-hand side there as well. And actually, you can see some of the lacy sort of networks that are adjacent to some of these twigs, if you will, as well. In the next slide, you can see some more of these twiggy bryozoans. This is probably from the upper Ordovician, I'm thinking, and so they're very common in the upper Ordovician, in fact, and so lacy bryozoans. In the next slide, you can see a cross-section, a thin section of what bryozoans, how they dwell, essentially, and so they would live in a certain, the zoecia would live in a certain tube, if you will, and then they would grow and cement the back part of that, and so you could extend farther and farther and farther. So that's how the colony would grow. And so you can see the open spaces there where the animal at least one time had lived. And so that's a very common life form. So the bryozoan has been enormously successful. They show up in the Middle Ordovician. And, of course, they today live in not only ocean waters but also in fresh waters today. And so you can find bryozoans in ponds and things like that, in fact. So kind of a very successful group that's made it around for a long time. The next slide shows you a mound-shaped bryozoan. There's one on the following page too. So here's we're going to go kind of rapid fire here because it's taking a lot of time to put this thing together. So anyway, so kind of a branch constellaria there. It's branchy, but at the same time kind of mounded, and so pretty odd sort of shape, I guess, on it. But Constellaria is pretty famous in the Cincinnati and around Cincinnati, where you get some of the upper Ordovician preserved. Very common fossil material. I know a specialist who works on Constellaria, actually. He's down in Harrison, Arkansas. Anyway, on to the next one there. On the lower right-hand side, you may think that you find a local starfish in the local rocks around here. They're pretty common, in fact, in the Burlington. So, and you think you've seen a starfish like that? Well, it's not really a starfish. It's actually a bryozoan. So it's called a Vectinopora here. And so a Vectinopora had the same shape as a starfish, but they would live on the seafloor as a benthic creature, having a biseriate sort of life plan, if you will, on each one of the extensions of those arms. And so it's actually a colony and not an individual starfish. And starfish are going to belong to the Echinodermata, which we haven't gotten to yet. So, but that is actually a type of bryozoan, not an echinoderm there. So, okay, so next on the list here are the brachiopods. The brachiopods, it's the phylum Brachiopoda, whereas it was the phylum Bryozoa for the previous group. Brachiopods go from the lower Cambrian all the way to recent. There's some questionable ones even in the Ediacaran, I think, that people think may be ancestors of the brachiopods. They're enormously abundant in the Paleozoic, and they still are alive today in some of the oceans, where they live on rocky shorelines and maybe under rocks and in crevices and places like that. But very typically, they are regarded as one of the common fossils that you will find in the Paleozoic then. So typically, if you pick up anything that looks like a shell in Mississippian and older rocks, you're going to say, well, look at this bivalve. And it's like, nope, that's not a bivalve, that's a brachiopod. So we'll look at the classification of these things a little bit, but brachiopods, once again, have that sort of bilateral symmetry that runs across the shell. So one side is the mirror image of the other side of the shell, and whereas the bivalves tend to have that mirror image in between the shells. Okay, so in between the valves. So, so these things really traditionally were called inarticulate and articulate brachiopods. And so the inarticulate brachiopods sometimes are called calciphosphatic brachiopods nowadays. And so that would include things like Lingula, which is enormously common in Cambrian age rocks and some lower Ordovician age rocks as well. And today they actually live, and there's a fossil. The fossil is called Lingulella, and the living organism that's still around today. So all the way from the lower Cambrian to the recent now. So Lingula lives in mud flats and things like that, where it's on the end of a pedicle. A pedicle is like an anchor, if you will, that's the foot part of these things. And it helps the brachiopod to stick out of the sediment interface, do its filter feeding during high tides, and then during low tides it'll draw itself back into its burrow. And so that's the inarticulata. That's the most common one, the living one today anyway, would be Lingula. The class Articulata is less common, in fact, but much more common in the Paleozoic, and they have calcitic shells, whereas the inarticulates have mostly phosphatic shells, so that's like a bony material, right? Calcium phosphate. Next slide, you can see a lingulid over here on the right-hand side. Now that's one that has been collected out of the mud, and you can see some living ones at the bottom down there. That's a phosphatic shell. They have little setae at the tips of their shell where the shell opens up, and so it opens up side to side there, I guess you could say. It's like it opens up like a trap, if you will, but that's the pedicle sticking out the back there. So apparently they don't taste very well. They have survived, the evolution of fishes and so forth. Human beings, I think, are the only predators for lingula, and so human beings in the Philippines will cook up the pedicle, and it's pretty tough sort of material, but that is an inarticulate brachiopod right there, or a calciphosphatic brachiopod, living ones today. And in the next slide, you can see some of the older ones. The Paleozoic ones are at the bottom down there, and you can see the difference in the symmetry. Again, the valve would open up towards you, I guess you could say, on the lower left-hand side here. And so, but the symmetry goes from right and left. It looks like a mirror image, right? When it comes to bivalves, there's one that's already open, right? And so they're showing you the plane of symmetry, in fact, is between the two valves. And so that would be, in the case of bivalves, those are actually mollusks, whereas brachiopods are not mollusks. They are their own phyla. And you can see how they've reconstructed the shell morphology. In the upper part there, you can see where the foramen is, where the pedicle would stick out of the shell. They would anchor themselves in the seafloor that way and then open up the valves and be able to draw water in currents into their shell and feed with the lophophore. And you can see the lophophore over here on the right-hand side reconstructed in cross-section of what a brachiopod looks like. And they have it flipped upside down, so the pedicle is actually heading out and up, whereas in reality it would probably be down in towards the bottom there. On the right-hand side and the lower right-hand side, you can see some living brachiopods. Those are pterobrachylid brachiopods. They live in rocky coastlines mostly, but they have them, they've been regarded as kind of like very primitive sort of organisms. They probably arose from worms like the phoronid worms. But in fact, they have pretty fast muscle fibers, apparently. So I have a friend who studied some of that, and they can snap shut actually much quicker than people would think. But generally, they keep their shells slightly gaping so they can draw in those ocean currents. The next slide is way too much information, but just know that we don't know exactly how to classify some of the organisms that are regarded as brachiopods, like the Craniacea or the Craniformia. It's listed on here, but they're calcitic in their composition, but they're lumped in together with the inarticulate brachiopods generally. So there are some controversies for brachiopods. I suppose I'm one of the few people on Earth that's ever taken a course called Brachiopod Paleobiology. My advisor when I was at KU was a brachiopod specialist. In fact, he wrote the chapter on inarticulate brachiopods in the treatise for invertebrate paleontology. He's a Brit, and he studied Craniacea for his master's thesis, as I recall. Actually, no, it was his PhD, actually. And he was a world specialist. He is a world specialist. He's just very old right now and very retired and enjoying the last years of his life, I suppose. And then I know some other folks that are brachiopod specialists as well. So there's one from Uppsala in Sweden, and I know another one who's at University of Kentucky, and Rebecca Freeman. And so Rebecca Freeman is also a specialist in these calciphosphatic brachiopods here. So on the next slide, you can see some of the fossil material. There's a Lingulella at the top, a lingulate of some sort. They can get up to like 2 centimeters in length or so. There's an orbiculid on the upper right there, and you can see some other ones. There's an acrotretid down here at the bottom. And the acrotretids are very commonly used as index fossils. Remember, index fossils have a wide geographic distribution, a short time range in which they existed. And so they're very amenable to being applied to the stratigraphic record for biostratigraphy, especially during the late Cambrian and actually even into the early Cambrian, early, middle, and late Cambrian, all the way into the lower Ordovician. They're pretty good for index fossils. In the next slide, you can see some more brachiopods here. These are orthid brachiopods. They have calcitic shells, so these are actually regarded. They would have been regarded as articulate brachiopods, and now they are calcite brachiopods. I forget what the large group is called now, so they have a new name for that as well. So, if they call these, the other ones, calciphosphatic for the inarticulates, I don't know what they call just the calcitic articulate brachiopods, but neither one of them talk as far as I know, so they're not really articulate in that sense, but they do open their valves up, and so that's the articulation point, I guess, for calling these things articulate brachiopods. In the next slide, you can see some bryozoans which have encrusted this shell, this brachiopod shell. So, here they are, 2 lophophorates in one photograph. And the next slide gives you the same sort of idea. In fact, there's even brachiopods on this brachiopod shell. This is a strophominid brachiopod, or strophominid. And so you can see the bryozoan is the crusty sort of lacy sort of texture on there, but then there's a small brachiopod on the very top of that shell. These things kind of spread out and would have rested on top of the sediment interface, the water sediment interface, and so you can see there, you get that idea that they may have been floating on the mud, essentially, on the seafloor. In the next slide, you can see some sporiferids. Sporiferids had a very elaborate calcitic sort of structure on which to hang their lophophore, and the atropods at the very bottom down here are just about the same, except they have a slightly different sort of orientation to that sort of support structure. So very specialized sort of like brachiopod right there. So that's the sporiferids and the atropods. On the next slide, there's one place in West Texas where many of the brachiopods there that are Permian in age have been silicified. And so what they had done in order to collect these was that they would take giant blocks of carbonate rock where these silicified brachiopods had been interstitial in that material, and they would lower those into vats of acid, and so the acid would eat away the carbonate and it would leave behind the silica like this. And so you get these elaborate sort of like tubes that ran off of some of these things. So that's a productive brachiopod right there, with the projections are actually openings. They have like they had like sensory sort of capacity to them, so they would be able to anchor themselves very, very carefully in the sedimentary succession that way. So that's pretty much the end of the brachiopods. There are some really weird ones that show up in the Permian as well that have, just one diminished valve, and then the other valve is just wide open. It's just, unusual, very weird sort of thing, but it's in a reef sort of setting. And so that's pretty much it for the lophophorates then. So we're going to move on next to the mollusks here. And so mollusks are our next major group, the phylum Mollusca. They're in Cambrian rocks. They probably are in the Ediacaran as well, but again, they're questionable sort of occurrences there. They have been an enormously successful phylum, and they are solitary animals almost exclusively, I guess I would say. Yeah, every bit of that. And then they make their skeletons out of calcite, aragonite. Some of them are chitinous as well. So that is, they have some organic material. And some have no skeleton at all. And those would be like the common garden slug, for instance, is a air-breathing mollusk. And so they've been enormously successful, not only from going from marine waters into fresh waters, but then from fresh waters into dry land. And so here we have the thought, in fact, that these things probably came from the annelid worms. Again, the annelid worms don't have much of a record, other than an trace fossil record, but would have been crawling around through sediment back all the way in early Cambrian time. In fact, that's what defines pretty much the base of the Cambrian, the earliest worm tracks and burrows and traces. So they have an organ level of complexity. It's much more complex and complicated than what the brachiopods were. They have a foot, if you will, a foot that allows them, a fleshy part, in fact, to be able to crawl. And so they also have a digestive tract. They have the true anus, actually. The brachiopods didn't have a true anus in their anatomy. They have well-developed kidneys and reproductive organs and so forth. The mantle, which grows around and secretes the shell, is a major organ in most in most mollusks, and of course they have gills, so the marine ones are able to extract their oxygen from seawater. So here are the classes. The Monoplacophora have a single shell, obviously. They were thought to have gone extinct at one point, but they recently discovered some, well, not so recently, but in 1952 they found some in the eastern part of the Pacific, and so they found living Monoplacophorans there. The Polyplacophora live along seashores very commonly. They're from Lower Cambrian to recent. They have plates on their back, essentially, and so the Polyplacophora are enormously successful as well, having gone from Lower Cambrian to recent. Rostroconchs are now extinct. They went extinct in the Permian. Rostroconchs look a lot like bivalves, except the shell isn't split. It's just kind of like a U shape, if you will, and so it didn't have the hinge line that modern bivalves have. Scaphopods still exist. They're called tusk shells very commonly, but they're tubular sort of shells, so that's the Scaphopoda. Bivalvia, which used to be called the Pelecypoda or the Pelecypods. So bivalves have been around since the Lower Cambrian all the way to recent here. And then cephalopods. Cephalopods have been around since Cambrian as well, late Cambrian time, all the way to recent time, and that would include things like the Nautilus, which has the shell, the spiral shell, and that was very common in many of the ones that were in all the way back to Cambrian time. They had a divided shell like that. And then, of course, that would also include the squids and the octopi, who are part of the cephalopods, which they've lost their shell, essentially, but they still have a hard beak that could be preserved. So the nautiloids are still around. They've been around since the Cambrian as well, to recent here. And the Ammonoidea are these ones that have these very elaborate sutures, or sort of like partitions, that would help them have neutral buoyancy and actually to resist the pressure from being in the deep sea, so their shells wouldn't collapse from all of the pressure. And so they have these very elaborate sort of dividers in their shells, and so that's the ammonoids. And they were from Devonian to Cretaceous. Now, and then lastly, we have the gastropods, which means stomach foot, if you didn't know. So they would crawl around and they would be able to pull things into their shell and eat it with a radula. They have like a tongue-like rasping device on them. They lasted from Lower Cambrian all the way to recent, of course, and that would include things like the common slug today. So in the next slide, you can see the thinking is that there would have been an ancestral monoplacophoran type of mollusk that existed that gave rise to each one of these classes of mollusk. And so in the upper part there, you can see Amphineura, I think it is, that's polyplacophoran there. There's the monoplacophoran off on the middle part on the left-hand side there, the bivalves, and then the scaphopods. And then as we go clockwise around this, there's the Nautilus shell there, so that's a cephalopod, of course, and then the squids have lost their shell. They're another type of cephalopod, and the gastropods down here at the bottom. So that's our hypothetical ancestral mollusk. That's what HAM stands for there, that acronym HAM. And next on the slide here's actually the monoplacophoran itself. That is the one that they found, Vima, I think it is, that's in on the right-hand side there. Neoplina, that's it, yeah. Neoplina. And then, and so that was found in 1952. You can see what the shell looks like right there. It looks like a bivalve without, missing the other shell, but there's no hinge line or anything like that. And so it's just a conical sort of shape like that. I'm going to go pretty fast through some of these. The next slide is the Polyplacophora. That's the Amphineura here, or a chitin species. And so that is, it has 8 plates on it, and so those plates are made out of calcite, so very tough animals, in fact. And so you'll see these things along rocky shorelines, like in tide pools and things like that, where they'll cling to the rock, and what they're doing is they're probably grazing on algae that are on that rock. And so, but they're, the mantle that surrounds them is almost like leather. It's very tough. So if you try to pry one of these, you're going to kill it if you try to pry it off of the rock, in other words. Very strong, muscular sort of animal that clings to rocks very easily in high energy sort of settings. On the next slide, you can see a Middle Cambrian version of what people think belonged to the Polyplacophora here, and that's the Wiwaxia. Wiwaxia was found in the Burgess Shale, which is a Middle Cambrian deposit from up in the Kicking Horse Platform up in the Canadian Rockies. And so these are actually phosphatic pieces that would have gone together and armored this sort of like worm-like creature that was there. So anyway, the thinking is that Wiwaxia is probably related to some early offshoot of, it and the Polyplocophorans share some kind of common shared ancestry, if you will. The next slide, these are kind of some also-rans out of the mollusks. There's the rostra conch right there, and you can kind of see how it looks like a bivalve, but it doesn't actually hinge or articulate in that upper part there, so it's more of a U shape. But they did have an excurrent siphon that would go out the back, and so the wastewater, as these things would try to excrete their waste, it would go out the back that way. In the next slide, you can see some scaphopods or tusk shells here. I think we have some of these in the collection, so you'll be able to see what a scaphopod looks like. These things have been around for a long time as well. They have kind of a secondary shell, I think, that covers the opening there. So they do have, I think, both two valves, but I'm not sure. So you might want to check on scaphopods if you went to like Wikipedia or something like that. I will note that if you look at the green background that shows the 3 or 4 scaphopod shells at the top on the upper right here, you can actually see a hole in one of those shells. That hole was made from a gastropod. So gastropods with their radula are able to eat things like bivalves. And so if a bivalve has a very circular hole where its muscles would have been inside of the shell, you can tell that's what the gastropods do. What they do is they rasp and open up. They drill a hole essentially into bivalves, and in this case they drilled it into the scaphopod and fed off of the scaphopod's musculature. And so these are Ordovician to recent here. In the next slide, we just have a little bit of an introduction to the bivalvia here. Lamellibranch is another term that people have applied to these things as well. 2 valves, obviously. This would include the oysters, the cockles, the clams, the scallops, the mussels, pectins, and so forth. And so these things have bilateral symmetry between the valves, okay, as opposed to across the valves, which is what brachiopods have. And there are marine and non-marine forms, of course. And so the common freshwater mussel is something that Dr. Chris Barnhart studies in biology. And so, so here's another organism that made it from saltwater to freshwater. And of course, they I guess, that's pretty successful when you can do that. And so there are a variety of habitats that they live in. And so, from deep water, relatively deep water, all the way to relative shallow water. And in fact, even at beaches, they'll burrow into the sand and so forth. There's one fossil group called the rudistids or the rudists who actually made reefs. And so there were giant reef-forming bivalves back during the Permian. Pretty fascinating sort of creatures. So they have a beak, or umbo, it's called. It's not really like a beak like on a bird or anything like that. It's just the beginning of the shell where it begins to sort of uncoil, if you will. And so that's the embryonic phase. And so they would grow outward, of course, from there. They have teeth on that hinge line that help them articulate between the valves and help them to open it up, kind of like a hinge. Well, the hinge itself is there. And these teeth are like protrusions that would allow like a ball and socket sort of articulation along that hinge. And there's various kinds of hinges that help us define the bivalves. They have a ligament that kind of holds the whole thing together, and so it's kind of like a spring on the outside of that hinge that keeps it shut most of the time. And then very carefully they can push it open essentially. So they also have huge muscles in there on the interior that will leave scars behind. The pallial line is where the mantle attaches, and you can actually secrete part of the shell that way if you're a bivalve. And then they have a pallial sinus where they also have a siphon to pull water in, and then they can push the water out that way as well. So in the next slide, you can actually see a diagram of what the foot looks like. It's for locomotion across the seafloor. You can see the 2 muscles there. You can kind of see other parts. They don't really show the siphon on this one although you can see where it would extend out perhaps there with the inhalant opening, they call it there, but some of these things have a long siphon as well. You can see that they have guts, they have nephridia, which are the kidneys essentially for these things, and a stomach, of course, by which they can digest the foods that they have in a sort of digestive tract as well. If you haven't had oysters, you probably should have some at some point in your life. Oysters are delicious little mollusks that we have, and also scallops are pretty good too. On the next slide, you can actually see pecten here, and pecten is one of the rare bivalves that actually has that sort of bilateral symmetry you can see here, and it's slightly asymmetrical, but I suppose you might be able to confuse that for a brachiopod, except pecten because it's the symbol of Shell, the oil company Shell, Royal Dutch Shell it is today. And so that shell sort of structure like that was very historical, actually, for the oil company, for the guy who first started it. His name was Marcus. What was his last name? Marcus Samuel, I think it is. And so Marcus Samuel, his father, collected shells in the Orient and traded in shells and shell-covered boxes and so forth. So that's the story of where the shell came from on the shell signs. And so that is pecten right there. So that's the one that is the symbol for shell petroleum. Here's the next one. You can see a cockle. That's the Atlantic cockle there. Some of these things have massive sorts of shells on them, can be quite weighty. Some freshwater mollusks in the next slide here. And they actually, some of them are endangered and some of them aren't. They're very common, I guess, in some streams, but not in every stream, obviously. And that's one of the things that we consider to be kind of the canary in the coal mine sort of thing for how clean are our streams, how fresh is the water in our freshwater streams. Here's in the next slide, you can actually see one of the giant bivalves. It's a giant clam from Australia. You always hear about divers getting their leg caught in one of these things. I don't know how quickly they close, but I suppose they can. So there's a big giant bivalve down there at the bottom. In the next slide, you can see scallops that are being prepared for a meal here. In the next slide, you can see an oyster that's on the half shell there. And so these things are all very elaborate bivalves that can be, in my opinion, quite tasty at times. And in fact, even the next slide, which is the class gastropoda now, we're actually talking about even tasty. They're even kind of tasty too, actually. If you have escargot, you're eating snails. That's a fresh air-breathing snail that you would eat if it's escargot, so probably related to that pulmonate snail there in some way. And so, look, they have eyes there. These things are amazingly intelligent, too. So mollusks are enormously intelligent. Now, the nudibranch lost its shell. Here you can see Turritella down at the bottom, kind of a high-spired gastropod. High spire meaning the angle of the shell it forms when it's in its spiral shape. Some of these things secrete a poison, and they have poison darts, essentially, with which to shoot fish or people that are unsuspecting. So cone shells have... some of the cone shells have poisonous darts in them, essentially, that you can, I don't know, it's enough poison to kill a human, so I've heard anyway. So you got to be careful, I guess, when you pick up a cone shell if it's still living and in the ocean. But they have very elaborate sort of patterns on their shells as well. Now, I was going to tell you, these things are enormously intelligent, and so octopi have this capacity to solve problems and go through mazes and things like that. And in fact, they can sneak out of their aquaria. There's a story of one at Monterey Bay Research Institute where they were missing some shrimp, and they were concerned that some of the help were getting in and eating some of these exotic shrimp that cost hundreds or thousands of dollars. And they put a video camera in there, and sure enough, they caught an octopus which had escaped from its watery aquarium, crawled across the floor, snuck underneath the two doorways. Apparently, he smelled the shrimp, got into the shrimp area, ate the shrimp, left the carcass behind, and then crawled back to its cage. Enormously smart sort of creatures, perhaps. And so the giant squids live in the oceans today. They are fed upon by whales, okay, the mammals that swim in the deep oceans to attack them, essentially. But the giant squids are, you always hear about a sperm whale or something like that has a giant squid in its stomach, perhaps. Back in the old days, they would find things like that when people were still whaling. Okay, so next on the view here, actually, you can see this time there's a bivalve on this next slide here that shows one of those openings that was created by a radula rasping and drilling into that cockle, essentially down here at the bottom, sucking out the innards and then leaving it behind. So that's the carcass of a bivalve down there. Some of these things are absolutely astonishing in their complexity of their shells. That's murex on the right-hand side. Murex was used by the ancient Greeks for purple dye, and so they would harvest murex shells and then in some way treat them to get the purple dye out of that snail. Snails at the top have the openings on opposite sides there, and so there's a propensity to have sinistral or dextral sort of like coiling in these things, and then they also have hyperstrophy, which would say, I don't know, it's just kind of a unusual sort of coiling habit, if you will. And so very interesting sort of creatures. So is it hyperstrophic and sinistral or dextral and, normal sort of coiling that these things would have? So that's what mollusks are called, malacologists. And so malacologists would study that sort of thing, perhaps. And so snails have this very unusual phenomena too. What separates the snails from the other mollusks is when they're in an embryonic state, they have a tiny sort of like larvae called a veliger. And in the veliger, it can maybe swimming around, and next thing you know, you see it snap, like snapping your fingers. The gut makes a twist in it, and that's called torsion. And so torsion is the one thing that snails have that all other bivalves do not have. And so that torsion happens instantaneously in the veliger. You can probably go to YouTube and find that, looking at torsion in a veliger. But any kind of neat that way. Here's some escargot on the next image. It shows you some more gastropods that are prepared with a little bit of butter and a little bit of garlic in there. Just absolutely stunning. So if you're interested in escargot, they actually have some at Jim's Steakhouse, Jim with a double M. And so the next slide, you can see an octopus. Also tasty, but not to be too carnivorous about this whole thing here, but the octopus can change its colors. There are other sorts of animals that do this as well, like the cuttlefish, which are also a type of cephalopod. So in other words, they can signal things, and they can actually make it into a camouflage as well. So if they're trying to hide from something, they can blend in with the background that way. In the next slide is one of my favorite movies when I was a kid, 20,000 Leagues Under the Sea. And that's Kirk Douglas there that's being wrapped up by a giant squid that's attacked the ship. It's a submarine called Nautilus. And so that's Nautilus. And so as you italicize the genus and species names in taxa, so too do you italicize the names of ships, as it turns out, right? So this is not the USS Nautilus because it didn't belong to the United States, because Captain Nemo was a citizen of the world. But that's the ship Nautilus there. In the next slide, you can see a special variety of cephalopod called a belemnite. Belemnites are famous for a couple of things. One of them is they have a guard in that upper part there. So that's the rostrum you can see there. It's called a guard once it is and actually I misspelled it on here. It's G-A-R-D. So not like guard, G-U-A-R-D. But a guard is the skeletal part of a belemnite, which is a lot like a squid. There's squid with a little tiny bullet in the end of it, I guess you could say, that helps them balance in the water, right? And so it's the pointy end of the belemnite there. Now, they are extinct, however, but even though they've gone extinct, They find these things in great abundance in the PD River in South Carolina. And there's so many of these things, in fact, that it became the standard by which people did oxygen and carbon isotope analyses. And so very commonly when people write the type of unit behind, so like parts per mil, parts per thousand, that is, of, for the content of the delta 13C or the delta 18O, you would also put PDB behind it, which stood for PD, the river's name, PD belemnite. And so V now stands for virtual PD belemnite. And so, or the Vienna PD belemnite. And so Vienna is where they do the analyses on the oxygen isotopes there originally. But there is a small v that they put in these things as well, and that stands for virtual. So they've been able to replicate the isotopic content of these PD belemnites, which seemingly have all the same values. So they compare it to it. It's a comparator. In the next slide, you can see Nautilus with its camouflage shell there, and then you also see some of the earlier versions of nautiloid cephalopods, and those things have straight shells, some of them. Some of these things could be up to 9 feet long or longer. And some of the ones that were even coiled could be like up to 6 or 7 feet across. So they're really quite amazing sort of like group of animals, and very, very, as I said, very, very intelligent. They use these chambers in order to like regulate how much air, and so they have some neutral buoyancy. And so you can put gas, their expelled gas can go back into the shell and cause them to float higher, or they can expel that gas and cause it to float lower, and so forth. And there's a connection that goes in between all of these whorls. That's called the siphuncle. In fact, there's a little opening that would have been a tube, essentially, that ran through the entire organism from the living chamber all the way back into some of the earliest whorls of that nautiloid there. So that is the modern cephalopod in one of the early fossil versions, and that's a diorama at the bottom down there that shows some corals, some rugose corals, and a cephalopod hanging around on the seafloor. So some of these were long and straight and narrow, and then some of them happened to be coiled like this, and some of them had very elaborate sort of U-shapes and so forth. Not so common, but the coiled one's very common, and the straight one's fairly common as well. In the next slide, you can see what ammonites look like. And so there are different kinds of sutures that you get. So the nautiloid down there at the bottom, which is Cambrian to recent, that's our nautilus that's living today. There were ones that compare to that. And you can see the goniatitic, and then the ceratitic, and then the ammonitic sutures that are on each one of these. And so in the right-hand side, you can see Baculites, and it has some ammonitic sutures on a straight cephalopod. So that comes from the Cretaceous of South Dakota right there. I collected some of that with my family back in the 1960s. So that is Baculites right there. And so in the next slide, you can see some of these sutures also. So you see the goniatitic, and then the ceratitic, and then the ammonitic sutures, and then you see one of those unusual U-shaped sort of cephalopods as well on the lower right-hand side. So just to show you that there's quite a lot of diversity in amongst the ammonoids here. So here's some other ones. In the next slide, you can see some of the unusual sorts of coiling characteristics. There's one that's even coiled like a gastropod on the right-hand side there, but U-shaped and open spirals and so forth. They were a little experimental in some of their shell designs. So that takes us through the mollusks. And so the mollusks are a really important group of organisms, very successful, and having went into a lot of different environments through geologic time. We're going to leave the mollusks and head on to the arthropods next. And so the arthropods are another just incredibly diverse, in fact, even more diverse, I suppose you could say, than what the mollusks were, because the arthropods include things like the insects, and the insects rule this planet pretty much. So the arthropods would include, that's the phylum Arthropoda, which means articulated joint or articulated foot, if you will. They go all the way from the Ediacaran to the Cambrian, and even into the recent here, obviously. There's a lot of described species of fossil material. over 1 million of those, I think. And so some of them are insects and some are arachnids, of course, and then the crustaceans. And you get some groups that are extinct, like the trilobites and so forth. But again, we're talking about solitary animals, and most of these have a bilateral symmetry. You could put the plane of symmetry right down through the middle of them, like a brachipod, mirror image on one side as opposed to the other. And so, at least in external appearance. They have a greater complexity again, so we're stepping up the complexity every time we go to the next level here. And they have an uncertain origin, but most people think they're out of some sort of worm, and maybe the annelids are a good place to say that there was a hypothetical ancestor from that group. And but they became armored at some point, and so these armored things crawled around on the seafloor early on. They have a head, a thorax, and an abdomen. And so in the fossil record, they can either be calcitic or they can be chitinous, and chitinous meaning that they're made of that organic substance, a lot like insects are, the crunchy insects. So they also molt. They have a propensity for like shedding their exoskeleton, running around like a soft-shell crab for a while, and then finally hardening up after a while. So they will grow that way, and so it allows the organism to not be stunted by the size of their exoskeleton. So in the phylum Arthropods, we're going to go into some of the more important fossil groups here, and that would include things like the trilobites. Now, that's the subphylum Trilobitomorpha, the shape of a trilobite, essentially. Trilobite means 3 lobes. And so there's a central lobe, which is axial, and then there's a pleural lobe on either side, which are symmetrical on the other side of the axial lobe. And so those are the 3 lobes that it refers to. But they also have this threefold sort of dimension as well, where they have a cephalon, which is the head region. They have the thoracic segments, which are the middle region. And then they have a pygidium at the very south end of the creature as well. So there's a threefold symmetry that way. Well, it's not really symmetrical except from side to side. But there's a threefold sort of division that way in the trilobites as well. So another major group would include arachnids, which would include spiders and scorpions. Of course, those are living groups, and some of those are caught in amber very occasionally, and so you will see some fossil arachnids from time to time, but not very commonly. But you do see actually fossil crabs sometimes. So that's this Xiphosaura, which is a horseshoe crab. And so horseshoe crab's fairly common. Pycnogonida are sea spiders, not too much of a fossil record, but so the ones that are in bold letters here are the ones that are pretty common. Euryptidae. Eurypterids are sea scorpions. They're now extinct, but they grew up to like 9 feet long, crawl around the seafloor, especially back during the Silurian. Myriapods, myria meaning many, I think here. There's a whole bunch of different like centipedes and millipedes and so forth. There is a fossil record of these things, but it's not very common. Again, you don't find them everywhere in the rock record, as opposed to, for instance, trilobites all the way up into the Permian. Pauropoda, and I'm not really familiar with some of these. Hexapoda, of course. You get the class Insecta, so insects are fairly common, of course, and Crustacea. You do get a lot of fossil crabs, however, not a lot of them, I would say, but certainly some. So that's the crustaceans there. Branchiopods, sometimes you get fossil shrimp as well in some special preservational circumstances. Rimmopedia, Cephalocaridiida, so a whole bunch of these things. And lastly, one of the more important groups down here towards the bottom are the ostracodes. And so ostracodes are a bivalved sort of arthropod. And so they actually open up the valve and you can see their little legs kicking and they'll swim in things like pond water. And so ostracodes have made the jump not only from marine to fresh water, making them very successful. They also have developed a protective armor around themselves, which is more effective than the multi-plates that you would get, for instance, in a trilobite. So that is the classification for the arthropods. Now let's take a look at some. In the next slide, you can actually see the thorax, with the cephalon covering the head region in a trilobite, and then the pygidium covering the tail region. And these things would articulate a lot like a pillbug would, or a a roly-poly, and so roly-polies would roll up if they were threatened. These things would roll up too if they were threatened, or being buried or something like that. So they have a propensity to try to survive in these situations. They had primitive eyes. The eyes were compound eyes like those of a fly, not surprising since they're both arthropods, but those compound eyes had lenses that were made out of calcite in the trilobites. Very astonishing that they would do that. When they shed their skeletons, they would follow the sutures around the cephalon. So in other words, they would break the cephalon apart, and then they would crawl out from underneath of it and wait for the next round, which is slightly larger, to harden then. The thoracic segments would be just shed then, and then the glabella, the pygidium would also be shed at the end. So there's a lot of different parts of these things. The glabella actually covers the stomach. The brain is thought to have been in where the backbone was. There's no backbone in these things. They're all invertebrate, right? But like in a roach, the main part of the nervous system would be under the upper part of the thoracic segments there. And so much like a water bug, essentially, here. So these things would crawl around on the seafloor, mostly grazing off of algae and things like that. And they would be predated upon by other arthropods that were actually larger, that would come along and try to snatch them up off the seafloor. Okay, so cephalon is the covering for the head region. The thorax, of course, is the middle part, and the pygidium is the tail area. The glabella is on the cephalon there, and that covers the stomach. And then the free cheeks would be perhaps spiny extensions off of the sutures off the side there. That's what free cheeks are. The pleural lobes is pretty obvious. The axial lobe is right down the middle. The hypostome was a little covering for the mouth region underneath of the trilobite. And then, of course, there were the eyes. And so these things were primarily, they were only marine as far as we know back at the time. So they went from Cambrian to Permian in age, and they could enroll. So there's two different major varieties. That's a polymeroid that you see here, meaning multiple sorts of like segments in it. The other type is called an ignostid, and you'll see an image of it on the next slide here. You can see an ignostid that looks like a little push-me-pull-you, with, in this case, a couple of extensions off the end of that fossil that show that you can tell the tail from the head. Otherwise, you may not be able to in these things, and they, too, would probably close up. Even though they had only a couple or maybe three thoracic segments, they could still close kind of like a clamshell, if you will, and be armored that way. It's been hypothesized, because you find these things around the world, the ignostids, especially during the late Cambrian and the early Ordovician, that these things would actually perhaps close around an ocean bubble, if you will, like a gas bubble, and be able to float around the world. And so they find them in sediment, however, and they found some of these things actually phosphatized and preserved in great detail in the Orston Shale. And so they have SEM images of all the appendages, and you can see these things, remarkable preservation in some circumstances. Orston Shales in Sweden there. And so they are one of the best index fossils for the late Cambrian, in fact. And so the late Cambrian is rife with these ignostids, and so these things are very useful for trying to determine where you are at stratigraphically. So they're like having a little pocket mass spectrometer with you when you go into the field, and you can open up a nodule, let's say, that's in a shale, and like, boom, there's an ignostid there. I was like, oh, that belongs to Glyptognostus reticulatus. Look at that. Yeah, I know exactly where I am at stratigraphically. I've been there. I've done that before, so it's really amazing to see people be able to do that. On the next slide, it shows you the family tree for these things, and so it just shows you the ones for the polymeroids as opposed to the ignostids. Now, the ignostids are the ones that we just saw on the last slide, but in the polymeroids, they both went extinct. I think that I'm not exactly sure when the ignostids went extinct, but certainly the polymeroids went extinct at the end of the Permian there. Anyway, just to show you that people are very people are world specialists, okay, in these things. In fact, I know some of the world specialists in trilobites as well as brachiopods. So, they run in the same crowd. So it's kind of interesting that way. In the next slide, you can see how elaborate some of the trilobites got to be with their spines and their projections off of their body. And so that's an unusual one down here at the bottom. I'm pretty sure that probably took months to prepare that specimen. But the more common ones, like the one up there at the top, I think that's Alrathia kingi from the Wheeler Shale in the House Range out in Utah. It's either Alrathia or Bathyriscus. But you see, most trilobites are not preserved that well. If you see a full trilobite like that, that means it's dead. It was died. It had died before it was preserved. When you see the bits and pieces, they just may be molt specimens, right? And so maybe it's just a piece that was like left behind from the creature that swam off to wherever it went to go harden up a little bit. Anyway, you see pieces on the right-hand side here, and that's the more common sort of form that you would see preserved, in fact. In the next slide, you can actually see one of these Eurypterids. And so Eurypterids belong to the Chelicerata here. And so that's a Eurypterid preserved in a limestone there down at the bottom. And you can see that it's made out of chitinous material. That's the darker material and only slightly calcitized. And so Eurypterids being sea scorpions, essentially, they grew up to 8 feet long, it says here, I guess. But in the next slide, we're dealing with some other Chelicerata here, and this would be a horseshoe crab, of course. A living one on the right-hand side, a recently living one on the right-hand side, fossil one on the left-hand side here. They're from Ordovician to recent, of course, but a very spotty record because they're not very common in the rock record. In the next slide, you can actually see the ostracodes. They look like bivalves, don't they? Yeah, except they have appendages hanging out of them and off of them. And so the bivalves could be closed up, and you can see one of the phosphatized ones down here at the bottom that shows you all the soft part morphology, which is much more complex than what's in a bivalve. But it is a bivalved crustacean in this case, so that's the ostracodes. And so ostracodes very common in the fossil record. So in the next slide, you can actually see a fossil dragonfly. And so that fossil dragonfly, there's actually places of extreme sort of like preservation of some fossil material. Those are called Lagerstätten. Lagerstätten. I don't know exactly what the term means exactly, but it's there's one in eastern Kansas at one of the quarries that's along near the Missouri border. In fact, it's not too far from the Missouri border. And it's a type of insect here that you see preserved in the rock. And so the animal would have drowned or something like that and then been buried with sediment. Unusual sort of circumstances. In the case of some of these unusual circumstances like this, it's either because of bad water that animals couldn't survive anymore and then got buried in very fine-grained sediment, preserving some of the soft parts. I mean, wings are soft. And so they preserve the impression of the wings and then carbonize part of the wings as well. And you can find them in amber, of course, as well, but also in siderite nodules and things like that as well. So these things are really unusual to find that sort of preservation. So Lagerstätten is an individual term, and then Lagerstätte are that wherever you find these things in that extreme sort of like depositional setting where you might preserve something. Okay, spider trapped in amber, pretty commonly you can like to sort these things out. So, okay, this is break time almost now. And for me, this image shows you a lot in one thing here, right? So can you spot the mollusks in here, and can you spot the arthropods? So, SpongeBob, he's a peripheran, right? So he's Periphera. Gary, his pet, is, of course, a gastropod, right? So Gary's a gastropod. He's a mollusk, right? And then we have a plankton. That would be up there with some of those microfossils that we first studied after we looked at the stromatolites. Patrick we haven't studied yet, but Patrick is a type of echinoderm. And so we'll look at echinoderms in a little bit more detail in a bit here. And then, of course, Mr. Krabs is a crab, and he's an arthropod, right? And then Squidward. Squidward is going to be a mollusk as well. So there's 2 mollusks in here, 1 arthropod, 1 sponge, 1 peripheran, that is, and then 1 echinoderm here, and then 1 protist down here. And then, of course, a mammal in there as well. So Sandy is the mammal. Okay, that's enough of that, right? So here's the list of here's the answer for all that. What do they belong to? Okay, so next on the list is the echinoderms, and the echinoderms have 5-fold symmetry. There's about 20 classes of these things. They go from the Ediacaran ? Into the Cambrian, and also all the way to the recent, of course, because you get sea urchins and starfish, and you even get crinoids, actually, living crinoids today in deep water. But they have 5-fold symmetry. Pentameral is the fancy word for that, so 5-fold. Penta meaning 5, right? So 5-sided or multiples of 5, in fact. So some of the starfish have 10 legs or 20 legs and so forth. You can get brittle stars as well in really deep water. Crinoids crawl around these days, although they used to be rooted in the old days. There's an extinct group called blastoids, which we'll look at very briefly here. And then also the sea urchins and the echinoids and so forth that exist. Mostly marine biohabitats. In fact, all marine biohabitats, in fact. So they haven't made that bridge or that jump into the fresh water that we've seen in some of the other groups that have already been around, like the arthropods, of course, and they've gone to land as well. And then also in the mollusks, where they've gone to fresh water and then to dry land as well. So these things are pretty successful, however, anyway. So things like the sea cucumber is still out there. And remember, you can chop the arm off of a starfish and it will grow back. So they have these sort of miraculous sort of like life plans that allow them to regenerate tissues. So technically they can live a really, really long time, I guess. Many of these things are actually made out of calcite. And so if we go on to the next slide, you can see the Stelloroidea or the starfish here. And there's the brittle star, which is the Ophiuroid here. And they go from Ordovician to recent here. 5 arms, of course, are multiples of 5 that we just talked about. They can crawl around, but typically they don't crawl around much. And so when they do crawl around, they use these sort of like tubes at the bottom of their feet to be able to grab onto things or grab onto bivalves and try to muscle open a bivalve, which is trying for dear life to hang on to and keep its valve closed so it doesn't get eaten by a starfish. They have what they call a lantern sort of structure, and it's a lantern that allows them to feed in the middle part, so a 5-fold symmetry, sort of hard mouth shape that will open up. And the tube feet that are on the bottom of these things are kind of like suction cups, if you will. But that's what they use for feeding a lot of times, to get things up to their mouth so they can feed on them. Echinoids are like regular or irregular, and here are some right in here that are from, I think these are Cretaceous ones here. But they're from Ordovician to recent, and then the irregular ones were from Jurassic to recent here, but those are from Texas, as I recall. In the next slide, you can see some blastoids. Blastoids were a lot like the ancient crinoids, except they didn't have the feathery arms that extended out. They only extended out through little slits around the top of their calyx. And so the blastoid calyx, relatively simple compared to that of the crinoids. But very similar to crinoids in the fact that they have 5-fold symmetry. It's essentially a sea urchin at the top of a stalk. And they would have anchored themselves to the seafloor. They went extinct at the end of the Mississippian, as I recall. On the next slide, you can actually see some living crinoids that have the feeding brachioles or arms, if you will, that allow them to filter feed. So they send these feathery appendages out into the current and are able to filter feed, and it funnels everything down to their mouth. These things are very successful. They kind of show up during the Ordovician, and I've seen some of the early holdfasts on some of these, for example, in the Ordovician, just a single stem piece which would stick to a hard substrate, essentially. So any sort of living creature on the seafloor could be encrusted by one of these early crinoids, eocrinoids they call them at the time, in fact. So there's a very complex sort of crinoid on the right-hand side where you can see all the brachioles, the arms that are appended to that calyx. The calyx is the head region, essentially, and so that's the part that would hold the stomach and so forth, and be at the end of the stalk and kind of direct the movement of the head into the currents and so forth, and be able to articulate the stem pieces that were down below to bend into strong currents, perhaps. In the next slide, you can kind of see a artist's rendition of what these parts are. And so you can see the arms of the brachioles that have the pinnules attached to them, which would be the feeding apparatus itself. And so that crown is called a calyx right there, and the columnals are below that. And you have a lot of parts on these things, of course, and as you might expect. So that whole thing is a stalk, and at the very bottom you get a holdfast down there that holds it fast to the substrate, whatever substrate it's on. Another group that's extinct are the rhombiferans. Very exotic, kind of unusual, and so we're not going to spend a whole lot of time on them here, but rhombiferans are very similar to primitive crinoids, I guess you could say, but I think they had six-sided plates on them, so in a rhombic sort of shape like that. So that is the extent that we're going to go into for the echinoderms, and so the echinoderms, that's Patrick's phylum there. So Echinodermata is the phylum, actually the full name of the phylum there. We only have a couple of more sections to go through here. Next we're going to go through the hemichordates, and that would include the graptolites and the conodonts. Then we get into the chordates, which are the fishes. And then we have a little bit on plants, 3 slides on plants, and then we finish the whole thing up. So I know it's taken a while to get through this, but thank you for your patience, and hopefully you're going to learn a lot of stuff, because fossils are really fascinating sort of things. And you learn a lot from them, and not only that, you learn a lot about geology this way as well. So fossils tell us a lot about depositional environments and other things. So graptolites. What are graptolites? Graptolites are mostly carbonized films, if you will, that would be hemichordates. They have gill slits and they have notochords. Well, they have a cord, a nerve cord, but no notochord. And so they belong to a class Graptolithena, and they existed all the way from late Cambrian time into the Mississippian. They're shaped like jigsaw teeth, so they're easy to spot in shales and other sorts of like fine-grained sediments. So, and in fact, I can tell you a little story, just a very brief one, in fact. I was on a field trip in Quebec, and we were looking at some shales with interbedded sediment gravity flows, some very fine-grained sediments that had shale, and that was a shaly consistency with pencil cleavage in it, so it's been tectonized a lot. But then in between were these grain flow events, which would be deposited from events from a shelf at some point. So this were like toe slope sort of setting, if you will. And then in those rocks, the field trip leader says, well, we're not quite sure what age these are, but they're kind of Cambrian Ordovician sort of age. And I bent down and I looked, and there's a graptolite there, and it's like, oh, well, here's a graptolite. It tells you it's Ordovician right there. Boom. So I wasn't being a jerk about it or anything like that. It's like, well, you want to get this identified probably, but that right here, this is a graptolite, so that tells you it's not Cambrian. In fact, the only graptolites that were around during Cambrian time were dendroid graptolites, and so these were actually discrete biserial colonies that were preserved in that shale. So if you know, who you know may help get you the job, but it's what you know that keeps you in that job, and people will respect you for that. So know a lot, I guess is what I would say. So those jigsaw sort of shaped teeth are what are distinctive about graptolites. And again, they're just like black carbonaceous films on the rock face, on bedding surfaces usually. They're originally chitinous, are thought to have been chitinous, and we don't know what they were, of course, exactly because they're extinct now. But the graptoloid graptolites, in fact, are the advanced forms, and they began during the early Ordovician. So there's like 1, 2, 3 stages just as you get into the early Ordovician from these graptolites. Those graptolites floated around in the ocean currents. And then when they eventually were incorporated into sediments, they would die and fall to the seafloor and then be preserved in that setting. So in the next slide, you can see one of these Cambrian dendroid graptolites here, kind of shaped shaped like a tree, right? So that's a dendroid graptolite. And the next slide shows you what they probably looked like when they were floating around in the ocean. And so they're not very big. I didn't have a scale on this, but they're maybe the size of like a silver dollar or something like that. But that's the dendroid graptolites. Not real common, in fact. The next graptolites are the graptoloid graptolites, and those are the ones like there's a pair of them essentially. There would be two colonies, and these things would have floated around the ocean, so like only two branches to a tree essentially, right? But these are the ones that were more successful. The dendroids went away, and then the graptoloid graptolites succeeded them. They're used as index fossils, and in fact, people wanted to establish the base of the Ordovician based on graptolites, whereas the conodonts probably have a much more accurate record, however. But as it turns out, the earliest graptolites kind of did help predicate where the base of the Ordovician was placed. So you can kind of see that not only were these things carbonized, they were actually cooked a little bit, so you can see that sort of whitish sort of material that's on the rock on the right-hand side on the bedding plane there. And you can kind of get the V sort of shape from these things as well, but these things floated around the ocean and were left behind and preserved in these bedding planes as they got compacted, smashes the colony essentially that would have existed, and then the theca, the living organism, of course, would have been dead in there as well, but that's what would have lived in each one of those individual saw teeth essentially, so the little animals, so it was a colonial animal. So on the next slide you can actually see the next group, which are conodonts. And we don't have a good example for them either in living animals, although we have analogs for what we think that they were like. They are chordates. They would actually have had a sort of a notochord that ran through the organisms. But the thing that gets preserved most commonly are these grasping sort of like appendages or teeth, essentially. They look like teeth. So they're kind of, that's where the name comes from, cone-shaped teeth, right? Conodonts. And so cone-shaped teeth were used for grasping or crushing their prey. And so they were predators, and or scavengers at least. The teeth themselves were either coniform or they're bladed. In other words, they have projections or platforms off of them. And platforms is another variety, in fact. And so these things very complex shapes, but very distinctive. And because they occur in carbonate rocks very commonly, you can dissolve the carbonate and pull out the phosphatic material. And that's what Dr. Jim Miller does, actually. He's retired emeritus from Missouri State University, a specialist in conodonts from the Cambrian-Ordovician boundary interval. So the individual teeth are called elements, okay? But the elements go together to form what we think is an assemblage or an apparatus of conodonts that would have been like the differences in the teeth in your own mouth, right? So if we have 32 teeth and you know, some of us lose our, you know, wisdom teeth or whatever, you know, but those teeth would be preserved, in fact, in the rock record. And so those are called elements when it comes to conodonts, and they tend to be very, very tiny. But because they're very tiny, they tend to be preserved. And so the apparatus is actually where you may see the entire assemblages of these things. Very rare that you would actually ever see that, although it was in the 1980s. Somebody discovered in Scotland in one of their museums is like, look at that. There's an assemblage of these things. And then after they began to look at the specimen, it's like, holy smokes, we got the whole animal. And so it was a soft parts impression of the animal that had this apparatus at one end. And so they were able to describe the conodont animal from that occurrence that had already been found and preserved in a museum. They're made out of phosphate, of course, phosphatic calcium phosphate, just like your teeth are. And so they were scavengers or predators, and they would grasp onto things or crush things that way. They were marine habitat only. They existed probably from the Ediacaran, although that's probably a question mark, but they certainly went into the Triassic. Now, the true conodonts didn't show up until like towards the end of the Cambrian. Before that, they were called paraconodonts. And because of that, it's probably a paraphyletic group. There may have been multiple things that had grasping sort of like teeth back at that time. So index fossils almost for their entire range, everything from the Cambrian all the way into the Triassic. People recognize what age rock they're in based on the conodont elements that are in there. In the next slide, you can see one of Dr. Jim Miller's slides of one of these things. You can see a white tip on the end of this. That would have been the part that was protruding from the gum or the mantle that was in this organism. And so just a simple cone-shaped sort of animal here. And this one happens to be relatively large, so that thing is slightly more than 1 millimeter in length, and that's unusual for conodonts. Did I say graptolites before? I meant conodonts. So that's a conodont anyway, a single element conodont that is a single coniform element. And so on the next slide, you can see platform and bladed elements. Platforms are more broad at the base, whereas the bladed elements are more linear in shape and like a serrated knife or something like that, right? So that's what you see at the top in this one, and actually at the bottom as well. Some bladed elements, and then also some simple coniform elements interspersed in amongst those. So these things grasp onto their prey. Here's an assemblage that somebody has reconstructed based on the collection of elements that they think that they found. On the right-hand side, you can see one of the rare sort of reconstructions that people did. Because if you don't know what an animal looked like, some people like try to make up what they think the animal looked like, and that looks like a toilet paper with a whole bunch of needles on it. I don't think, even that was never accepted by most conodont workers back in the day, although it was published that way. And anyway, it was kind of rejected after they actually found the animal that had the assemblage in its mouth region. They look something like the middle image in there, where you can see something that's kind of like an eel sort of shape there. In fact, that's the modern analog we use today to try to figure these things out. And at the very bottom down here, you can see an assemblage of these things as well. So the actual animal probably looks something like that middle sort of reconstruction for the conodont animal there. Big eyes for a predator, and then the grasping teeth in order to grab onto whatever it's wanting to eat. So their reconstruction, very commonly people will use hagfish, which is on the right-hand side here, to say that's probably what the conodont element was placed in the mouth of an organism that looked a little bit like a hagfish. Now, hagfish are disgusting animals, I got to tell you. They are slimy, I think is an understatement. They have a tendency to grab onto fish and then eat their way into the fish, and then live in the fish even after it dies, and then come out of the fish finally when it's finished feeding. So they find them in the Great Lakes, for instance, in fresh water today. On the next slide, you can actually see a hagfish that people are disgusted by. Notice they have the gloves on here and the thing wiggling around. And so that's a weird sort of thing. But here you can see some folks eating hagfish on the lower right-hand side in that little inset image right there. So that's what people kind of think the conodont animal was like. And of course, it was a little bit smaller probably too because of the size of the elements. So that's all about graptolites and conodonts. Next, we're going to go on to the fishes. And so finally, we're in the chordates here. The next image shows you, there's a famous artist. His name's Ray Troll. Ray Troll does T-shirts and all sorts of things. In fact, he wrote a book called Cruising the Fossil Highway. He's done a couple of those, actually, so I think it's Cruising the Fossil Highway 2 is out now. And he does the illustrations while there's a paleontologist that he travels the West with, and they go to various places that do excavations of dinosaurs and swimming reptiles and that sort of thing, and fishes like the Xiphanthes down here at the bottom that has a fish caught in its mouth. So he's a kind of an artist who's a pedestrian sort of like illustrator, a cartoonist essentially for fossils. And so that's Ray Troll's work right there. And you can see some of the fossils that he's shown before. You can see on the right-hand side, there's actually a sea scorpion or a eurypterid on the right-hand side. And then there's it's not a mosasaur, that's going to be a plesiosaur, I guess. And the second one over, and then a big turtle, a kilo something, I can't remember the name of it. Big sharks behind the megalodon, and then of course another swimming mosasaur over here that has its muzzle strapped in. So these deep sea fishermen here are bragging about the catch that they have. And of course there's the big salmon right there that they probably actually caught, but showing you all the things that went into making that salmon, or how the salmon gave rise to many of these other things that were out here, in fact. And so ichthyosaur in the middle ground there, and then down below here is like looks like Tiktaalik down here on the right in front of the fisherman with got his boot on top of the head of Tiktaalik there. That's the early amphibian, of course. There's some plated fish in here as well, so the placoderms, and then there's also these fishes down in the front here, so another mosasaur. Anyway, kind of neat illustration there. So the chordates. The chordates are a group that came together as a phylum, and so we're a part of the chordate phylum, if you will. And the fish are part of this, and they've been, there's several different classes of fish, and so the agnathids are ones that are jawless fish, and so they would have existed all the way from Cambrian time. For instance, the lampreys and the hagfish are actually a variety of the agnathids, I think here. The acanthodians are spiny fins, and so that's how people differentiate these things based on their characters, right? So Silurian to Permian for them, a sort of combination of chondrichthyes, which we have as like a shark, right, as a chondrichthyes type of fish, and then the osteichthyes, which is the bony fish. And so acanthodians kind of like in the middle part of there. Placoderms are the plated fish, if you will, so armored with their heads, and the jaws are very tough, I guess you could say, and then but the jaws, and they have plates all over them. Salernian, Devonian in age mostly. Chondrichthyes here you can see again. That's the shark skates and rays. Cartilaginous in their composition of their backbone, essentially, but the osteichthyes with bony backbone, of course, and rib bones and so forth in them. So they are from late Silurian to recent, and that's what most modern fishes are. Now, there's one offshoot of the osteichthyes that are really pretty important, and that's the Sarcopterygians, or the lobe-finned fishes. And they were found first as fossils, and there's just a couple of them that are around today. One of them is a lungfish, which is early Devonian to recent here. They actually live in fresh water and burrow into muds, in the idea that, if things were to ever to dry out, they would come back again when the rains came back. And so that's what lungfish are. In fact, they can live in the ground for a while until they get wet again, and they will come back out into the rivers. And that's mostly in Africa, I think, the lungfishes live. The other one is the coelacanth, and the coelacanth is an interesting sort of fish that was seen in fossil material first, lobe fins on it, and then they caught one in the 1930s, and it was caught in a net off of South Africa, and so they brought this thing in. It's like, what is this thing? And they got it to the right person, and this person, I can't remember her name, but she reported on it, and it was like catching a fossil out of the ocean. Today, they find coelacanths around many of the islands in the Indian Ocean, but actually still off of the coast of South Africa as well, in deep water. And so they find the coelacanths in that sort of situation. Now, I've got some images of a coelacanth here. And then also you find fossil fish, even in some settings, in freshwater. And so even in the Mississippi and even in the Paleozoic. The next slide gives you some indication of how the family tree goes for the Astracoderms, the agnathid fishes, of course, and then the chondrichthyes, and then the osteichthyes, and so forth. So the chondrichthyes, the osteichthyes, and of course the agnathids with the hagfish and the lampreys and so forth that are extant even in the Neogene, even in the present day today. So here we have the agnathids. There's our hagfish attached to, or lamprey in this case, attached to a salmon there. And you can see their sort of shape. And you've already seen what the hagfish looks like up close and personal there. The next shows you, the next slide here shows you the astracoderms. They're an extinct version of agnathid. And they have scaly sort of like covering on them, but they have a kind of a tough head on there as well, but still you find them in fossil material sometimes. Acanthodians are now extinct. That's the next slide here. Man, there's a lot of fish out there, so I can't really tell you what really distinguishes some of these from other ones, other than they have bony spines on their fins. And so you can kind of see the spines projecting off of the fins there. In the next slide, you can see a placoderm. Now, placoderm's easy to describe because you think about plaques and plates essentially, right? And so this has plates all over its thorax and its head here. And so that's the placoderms. And they did not give rise, in fact, to what looks to be like a turtle down here on the right. That's actually a placoderm fish right there. Now, the earliest fishes, as it turns out, probably had a light-sensitive spot in the middle of their skull. And that would be, I think some of these placoderms had that to begin with, but then they evolved eyes after that. So eyes have evolved in living organisms in many different patterns, in different ways. So they have different components, in other words. On the next slide, you can see a chondrichthyes, a cartilaginous fish. You can see that it has a bony spine on it. Doesn't mean they can't have bones in them because, for instance, even in sharks, they have bony teeth, right? If they were all cartilaginous teeth, we wouldn't worry so much about swimming in the ocean, right? But they have a cartilage for their cord, their nervous cord, and but they have bony spines sometimes as well. In the next slide, you can see some more chondrichthyes here with the bony spines, in fact, and so you can see some of these fish preserved, again along bedding planes and so forth. But it's the bony material that tends to be preserved, and sometimes the cartilaginous material is preserved as soft part or kind of fleshy material that are preserved like carbonaceous sort of film almost. In the next one, you can see some of the teeth that are associated with sharks. Some of the earliest sharks, in fact, were not carnivorous or even scavengers so much, but they were shell-feeding. Well, I guess they were still predators, but they would eat shellfish for the most part, and so they would develop teeth to crush things with. And some of them actually had like a raspy sort of appendage as well to cause them to be able to rip things to shreds, essentially. And the next slide's another ray troll image of one of the sharks, the chondrichthyes, around playing cards there. Card-playing chondrichthyes. And then the next one, of course, is the more common modern fish that we have today, and that's the osteichthyes, again with the scales, but then also the bones. And so the bones are what tell us that it's an osteichthyes, including billy bass, of course, and so bass are a type of osteichthyes. In the next slide, you can actually see a Sarcopterygian here. Now, its lobe fins are not quite so developed as some of the ones that you'll see later here, like in the coelacanth, but you can tell that they are kind of lobe-shaped, at least in the reconstruction of what the animal is at the top there. And the next slide shows you somebody diving with a coelacanth. That's a living fish right there. Latimeri. It was named for the person who discovered it, I think, and so discovered in 1938 in a fishing net, but then brought in, and I've got a video, I will link to this from, I think it's EOS or NPR or something like that. So it's a public television video that you can go take a look at to see the story about catching the first lobe-fin fish off the coast of South Africa. But there it is in all of its glory right there in its living environment. So the lobe-fin fishes are really, really, really important, especially to us. And so because of them, we probably developed amphibians. And so Tiktaalik is thought to have developed from the lobe-fin fishes, for instance, Tiktaalik, the genus. They found it in Greenland maybe 10 years ago, 15 years ago, 20 years ago maybe. But anyway, here's some of the earliest amphibians that developed back during the Devonian. That's Tiktaalik. It's the earliest known one anyway. And some of the lobe-fin fishes that existed back then. So these lobe-fin fishes were around back in the early Devonian. And so it's a pretty important story. So they made it all the way to recent time, but in fact their greatest success, you might say, is being able to give rise to the amphibians who laid eggs in shallow water, but were able to crawl on dry land. By being able to crawl on dry land, the amphibians gave rise then to the reptiles, and the reptiles had amniotic eggs. And having amniotic eggs, they were able to lay their eggs on dry land where they were safe from predators because there was nothing around to eat them back on dry land back then. And so they would be, and this is during the Carboniferous, and so they would crawl back and swim around in the ocean if they could. They're amphibian. Well, the earliest ones were amphibious, I guess you could say. And so that's the early reptiles. And some of them stayed on dry land, right? And so some of them would give rise to like Dimetrodon back during the the late Carboniferous and into the Permian. They gave rise, in fact, to animals like Lystrosaurus, you may call, or Cyanonathus, and those sort of animals gave rise to mammals and were a mammal. And the Dimetrodon gave rise to other reptiles that made it across that Permian extinction, as did the early mammal-like reptiles that gave rise to the mammals in Triassic time. And in Triassic time also the dinosaurs evolved. And so it's all because of this stepping out onto dry land. So the lobe-fin fishes we owe a lot to, I guess you could say. So in the next slide, here's the amphibians, the tetrapods. They have 4 feet, in other words. So and next to them, the lobe-fin fishes that are really important to us. Another ray troll sort of image right there. So that wraps up the animals as far as we're going in historical. Thank you very much for giving rise to us, you mighty lobe-fin fishes, Sarcopterygians, you. And then we're going to go briefly into the plants because plants are around, and you will see some of these actually even in Missouri rocks. So with the plants, we have things like Lepidodendron here. That's the image in the middle that shows you the bark that was on the outside of essentially what's a scale tree. So some of the earliest plants probably came around in Devonian time as well. When the mammals came, the lobe-fin fishes came on shore, they probably arrived to an area that was relatively devoid of any sort of land cover. It was just sediment for the most part at that time. There may have been some living plants there, but there's not a great record of it before the Devonian. There are some fungi that people think that they have found, however. So as plants go, most of these are either carbonized or they may be silicified as well. So silicification is another way to preserve, or permineralization, like with fossil wood, right? You permineralize it. They're abundant land fossils that began in mid to late Devonian time, however. So there's a certain amount of coevolution that's going on between animals and plants at this time. So in the plants, there are the tracheophytes that are vascular plants. There are Psilopsidids, and then there's Lycopsidids. So all of these things like club mosses and ancient scale trees, for instance. And so those things would have been common even in the Pennsylvanian. So in the Carboniferous, these things were around. Sphinopsids in here. Calamites is one of those. So I think they call it snake grass. It's a lot like snake grass, in fact. At least it has that sort of texture. Some of the other sort of organisms that came around, we finally begin to have simple forests back during Devonian time, and that would include like the fern trees, or the seed ferns, in fact, even began a little bit earlier in the early Carboniferous. And so we find some fossils of ancient seed fern trees, even locally around here, in some of the marine deposits, actually. So they would have been driftwood that fell to the seafloor and became silicified along with all the other chert that got silicified back then in some of the Mississippian carbonates around here. And so those are some of the earliest seed-bearing ferns. They call them sperms, pteridosperms, I think it is, pteridosperms, the early seed-bearing ferns. Now, most modern ferns are not seed-bearing, but there were ferns all the way back to Pennsylvanian time. So ferns have been around a long time, let me put it that way. They gave rise to a group called the gymnosperms. And so when we think of gymnosperms, it's easy to imagine them. We're really talking about cycads. Cycads are a type of, almost looks like a pineapple. It's not a pineapple because the pineapple grows at the top of the stalk. But, these are things that had fern-like leaves that shot out the top of something that was pineapple-shaped. Ginkgo trees. Ginkgo trees are absolutely ancient. Those things that are like Ginkgo biloba, right? You're familiar with that, right? And then also like conifers. Even the early conifers show up at the end of the Paleozoic here. And then, of course, the seed ferns on there as well, the pteridosperms. The angiosperms finally came around during the Mesozoic. So the angiosperms would include all the flowering plants that we have today, right? So the oak trees and all the common garden plants, the fruits that we eat, the vegetables that we eat, all of those things came out of the angiosperms, the flowering plants. And they really made their presence known after the KPG boundary impact. And so they existed before that in the late Cretaceous, and maybe even in the early Cretaceous, the angiosperms were around, but they weren't abundant, and you very seldom found any sort of fossil record of those things. But they did show up. And even the grasses are, of course, related to these things. They put on flowers, essentially, right, and seeds. So that's as far as we're going to go, almost into the plants. I'm going to show you some pictures next so you can see there's a fern in the upper part there. There's a gymnosperm, right? There's a ginkgo leaf there that's preserved as fossil material. You can see it on the right-hand side in the living leaf there. And then there are things like the bryophytes. So actually the bryophytes I don't have on here, but I think that's the earliest angiosperm that's ever been published right there. It came out of China. But then here is a leaf on the right-hand side, and so that's another type of angiosperm as well. So most of the trees are not gymnosperms. They're the angiosperms instead. So those are our plants for as far as we're concerned is in this. If you want to learn more about that, you can study paleobotany. But as far as paleontology, if you're interested in that, you should probably take Damon Bassett's course on invertebrate paleontology, and you'll get everything that we covered in this one lecture for an entire semester. So that's probably an easier way to make it more palatable. But anyway, it's pretty important stuff, even for you, because the more that you're exposed to, maybe bits and pieces will actually stick. Let's just hope you're accurate in placing those memories, and that I'm accurate in actually conveying the information to you. So that gives us a story of life as it's preserved in the rocks as fossils. You know how fossils are preserved. You know the various types of fossils ranging all the way from early Precambrian times in the Archaean time, right? So almost 3.5 billion years ago, life has made this enormous flourishing, if you will, even if it had been punctuated by periods of extinction. So life, everything that we know of on Earth is either made out of DNA or RNA. That's how you have to replicate things. And so the DNA and RNA tell us that everything on this planet has to be related in some way, unless it came from outer space, of course. But even then, we've never found RNA or DNA in outer space material. Although we do find some of the earlier precursors to RNA and DNA in the form of amino acids in some of the meteorites. So maybe life did come from outer space. Well, we'll talk about that some more in class here, I think. That's a good topic right there. Even things that are the most primitive, and you got to wonder, is that thing alive or not? There are things called prions, and prions are the things that are responsible, for example, for mad cow disease. That's called a bovine spongiform encephalopathy. That's the disease you get from these things, these prions. And so even with those things, they even have RNA. So, even viruses have DNA, or at least RNA, right? So even the coronavirus that we're enduring this pandemic right now has RNA to help it replicate. And so that's how it does it. It does it with your help or whoever is infected with that virus to be able to replicate within your body or within whoever's body who is certainly infected at this time. So you have to have a host even with those things, but those things are alive at least while they're in their host. So replication and metabolism seem to be the key features of life. You have to be able to replicate, and you have to be able to make energy out of the substances that are around you. Prokaryotes are probably some among the earliest living forms. All creatures need energy to survive. We can get microbes even deep inside of the earth. And so you'll find microbes even in oil reservoirs in the deep 25,000 foot oil reservoirs, let's say in Oklahoma. They find microbes eating some of that oil down there. So amazing how evolution finds a way. Who was it? In Jurassic Park, they said life finds a way, and that's true. Jeff Goldblum, right? His character. So fossils are that record that go back at least 3 billion years, maybe 3.5 billion years. It's a lot of material we covered in here, but hopefully it gives you a time frame in which to organize your thinking about how fossils are found, where they occur in great abundance, that is during the Paleozoic, then afterwards. after the earliest origins. So that's it for now, and I'll talk to you later. Bye now.